morning to our respected Pro Vice Chancellor Sir, respected Dean Sir, our Chief Guest, Guest of Honor, and all our delegates present over here. My name is Joyita Bhattacharya, Assistant Professor, School of Medical Sciences, Adamas University.
And I am Srija Shur, Assistant Professor, School of Medical Sciences, Adamas University. It is a huge privilege for us to welcome you all in the third National Conference on Healthcare Industry Academia Summit 2022 and beyond. The conference is collaborated with Central Ayurveda Research Institute, Ministry of Ayush, Government of India.
This conference aims to provide an excellent platform to acquire the knowledge on the recent trends in pharmaceutical discipline through the oral, poster presentations, plenary and keynote sessions. We are eager to hear the enriching scientific talks of our distinguished speakers that would not only transform the event into a multidisciplinary platform for innovative ideas, but also kindle a sense of encouragement to undertake productive research work.
Prayers works wonders. Prayer should spring from the heart. To begin this day orderly, let us first invoke the presence of Almighty with a song. The song is being presented by the faculties and students of our school. Hello.
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 Masa
thank you for the heavenly aerials for letting the goodness spark to the spirit chain reactions with your angelic voices.
Light symbolizes brightness and prosperity. It removes the forces of darkness and gives us positivity. With this noble thought, let us start this occasion by lighting of the lamp. I would request our dignitaries to please come up on the dais for the lighting of the lamp ceremony. Sir, please. I would request our Pro VC sir, our Dean sir, Chief Guest, Guest of Honor, and Rodit sir, and our convener to please take up the seats on the dais.
Today, we have amongst us Professor Dr. V. Ravichandran, Director, NIPER, Kolkata, as our chief guest. It is our honor to have you amongst us, sir. It is an honor for me to welcome you on behalf of School of Medical Science, Adamus University. I would request our Honorable Pro VC, Professor Dr. Jitendra Pandey, sir, to kindly felicitate Professor Dr. V. Ravichandran. Sir, please.
Thank you so much, sir. It is a pleasure for all of us to have the eminent personality, Professor Dr. Ronindra N. Shah, former director, BITS Pilani, Dubai campus, as the guest of honor for today's conference. Heartiest welcome to you, sir. I would request Dr. Roger Tre, Professor Emeritus, to kindly felicitate Professor Dr. Ronindra N. Shah. Sir, please.
We also have amongst us our second guest of honor, Dr. Anupam K. Mangal, Assistant Director in charge, Scientist 4, CCRAS, Ministry of Ayush, Kolkata. He will be joining us through online mode.
Now I would request our Dean Sir to kindly felicitate our Honorable Pro Vice Chancellor, Professor Dr. Jitendra Kumar Pandey. We welcome you, sir, and thank you for your precious time. It is a pleasure for us to have Dr. Alok Ghosh amongst us. I would request Dr. Roger Tre to kindly felicitate him. Sir, please.
We have amongst us Professor Emeritus Dr. Roger Tre. So I would request our respected Dean Sir, Dr. Shajul Kumar Jha Sir, to kindly felicitate him. Sir, please. Like the rainwater brings life and prosperity to land, your words bring enthusiasm and strength to our hearts. Let me take the opportunity to call the backbone of our school, our respected Dean Sir. I would request Professor Dr. Shajul Kumar Jha Sir to kindly address the gathering with his motivational words. Sir, please.
Good morning. Good morning. Hello. Yeah. Distinguished dignitaries on the dais, off the dais. Special thanks to Professor Ravichandran sir, Professor Saha sir, Professor Ghosh, our mentor, Professor Ray, always supportive, our Honorable Pro Vice Chancellor, Research and Development, Professor Jitendra Kumar Pandey, Head of the Department, Dr. Sanjay De, Convener, Dr. Nandan Sarkar, and the joint conveners, Anesh Sabarik and Susmita. All other colleagues, members of scientific and other committee members, I heartily welcome to this third National Conference on Healthcare, Industry and Academia Summit 2022 and beyond. In collaboration with Central Ayurveda Research Institute, Ministry of Ayush, Government of India, the aim of the conference is to understand from different academicians and industry personalities regarding our healthcare research. We have received almost 200 posters, scientific posters, from 30 different colleges across the country. And the scientific committee members, they have done a tremendous job, and we have selected 122 posters. We have received around 30 for oral presentations. So great response, and I want to thank each and everyone, my students, volunteers, everyone. You have supported like anything. And last but not the least, the management of Adamas University, Honorable Chancellor sir, Vice Chancellor sir, and the team for their continuous support to make this event a successful one. Thank you one and all.
Thank you, sir, for your continuous motivation. Your words means a lot to us, sir.
If your action inspire others to dream more, learn more, and become more, you are a leader, says John Quincy Adams. At this juncture, I would like to attribute this proverb to our Honorable Pro Vice Chancellor, Sir, Professor Dr. Jitendra Pandey. We welcome you, sir, and would request you to kindly address the gathering. Sir, please.
Good morning, everyone. Respected Professor N. Ravichandran sir, Professor Saha sir, Professor Ghosh, Dr. Sarkar, our mentor, Professor Rajat Ray, and one and only our Dean, Professor Sajal Saha. You know, this summit, the third Industry Academia Summit, I think this is one event that is not limited to poster presentations or oral presentations or inaugurations. Here we come up with some actions, we come up with some plan, and we try to inculcate, try to include those action and plans for the betterment of our students. Sir, Adamas mean uncut diamond. We are nearly 130 plus acres of campus. And whatever we do here, sir, it's always one thing in mind, that how our student will get benefit. So today's summit and today's event is also for the same thing, that how we can train yourself, how we can plan better way that you can be prepared for the future generation or once you complete your course from Adamas University. We are grateful to have such renowned personalities among us. So my request to all of you, at your age, all questions are great questions. So do not feel shy in interacting, meeting and asking some questions from Ravichandran sir and Saha sir and Ghosh sir. It's a very rare moment for you. Ravichandran sir is, the way I know him, he is one of the very busiest person with lot of responsibilities. So one thing you can learn from him, how to keep calm and cool even with so many responsibilities. So thank you so much, sir, for coming. Saha sir, thank you so much. I mean, I can feel the energy, the positiveness when we meet from you, and I'm sure my students and faculty members even will learn a lot. And you have already been interacting with the one and only, once again, our mentor, Professor Ray sir. He has been with you, among you. So sometime you will feel like he's your mentor, he is friend, he is philosopher. So he is almost everything for the school and for the students. And Ghosh sir and Sarkar sir, they all are doing great. And last but not least, please put your hand together for Dr. Sajal Jha. He has been working 24/7 in real sense to make events, to make life of students better. So thank you everyone. Enjoy this event, enjoy this conference, try to make some notes and try to ask some questions. Thank you so much. All the best.
Thank you so much, sir, for your kind words. Now I would request our second guest of honor, Dr. Anupam K. Mangal, Assistant Director, Scientist 4 in charge, CCRAS, Ministry of Ayush, Kolkata, to join. I would request the media team to kindly help with the same. He will be joining us through online mode. Sir, please kindly address the gathering.
Okay. Am I audible to all of you, sir? Yes. Hello?
Yes, you are audible. Maybe we are not audible.
Excuse me, can I audible?
Yes, sir. Yes, sir.
Okay. Respected Dr. Jatendra Kumar Pandya, sir, Pro Vice Chancellor, R&D. Associate Dean, Dr. Sajal Kumar Jha, sir. Dr. Professor Ravi Chandran, Director, NIPER, sir. Dr. Raninder N. Saha, former Director, BITS Pelani, Dubai Campus, sir. Distinguished guests and delegates. Ladies and gentlemen, first of all, I thank the organization for inviting me to this summit, which has a very appropriate title, that is Healthcare, Industry and Academic Summit 2022 and beyond. This topic is very relevant in present scenario, as we all know that when biomedical research and its forward march is becoming increasingly dependent on academic industry proximity, means with industry as well as with academicians who have to do a lot of work. So we now have the potentials to enter one of the most productive periods in biomedical research, the success of which will depend a lot on an increasingly close partnership between industry and universities like ADAMAS and other universities. We all understand that economic partnership between industry and academic accelerate medical innovation and enhances potential access to the medical advances, which is the need of the hour today. Most clinical studies that bring new drugs from bench to bedside are financed by pharmaceutical industries or companies. Many of these drugs trials are rigorously designed, employing the skills of understanding clinical researches at leading academic institutes. I was reading somewhere and came across some beautiful lines which said that within many hundred years, time when people will reflect on history, the 19th century might well be written as the century of industry, and the 20th century as the century of information and technology, and in the 21st century as the century of biomedical and healthcare. At the present time, when universities are looking for funding these industries, funding is supposed to be help disease prevention and treatment, improve clinical practices, and results in useful products for patients. Without industry fundings, improvement advances in disease prevention and treatment would not have occurred. However, on the other side, a number of important areas of, of the connect between academic and the medical professionals and the pharmaceutical industries have been highlighted in the last decades, especially in the last 5 years. As you also know that we are working for Ministry of Ayush, and we are having our legal document Ayurvedic Pharmacopoeia of India, when protocols or monographs are written in the case of in these pharmacopoeia, we are associated with all the industry, herbal industry, and we will take their views, what they want to say in that one protocol. After, you can say, harmonization with the industry, we are preparing these monographs. So the results or the outcomes of these monographs will be acceptable everywhere, not in India, even is in a global level. I mean to say, the outputs or the work which industry will suggest to us, we will add data in our protocols or in our monographs. Our obvious concern is that academic industry ties should not bias research, both the kind of work that is done and the way it is reported. Researchers might understand studies on the basis of whether they can get industry funding, not whether the studies are scientifically important. That would mean more research on drugs and device and less designs to gain insight into the cause and mechanism of diseases. It would also skew research towards funding trivialist differences between drugs, because those differences can be exploited for marketing. Of even greater concern is the possibility that financial ties may influence the outcome of research studies. So we will need to look at these aspects also. So we can discuss a lot of issues on these issues in these type of workshops, seminars, or you can say summits. Well, there are some problems as an inevitably with all such potential controversial but useful relationship. Rather than concentrate on and magnify the faults, it makes more sense to ascend the positive and create an atmosphere whereby positive continues to be maximized. One positive outcome of academic industry tries would be that will add to the commercial value of academic innovation and help sustain it in the long run, as well as positive great window to opportunity to talent coming out with academia. So I will also want to tell you these things that now our council is having doing lot of project works in the form of extramural research and you can say intramural research also. We are doing a lot of collaborations with all the research organizations such as DST, DBT, CSIR, NIPER, and all. Presently, we are also having collaborative projects with NIPER and IITs also. So this is the platform for us that we can collaborate with each other. So the funding, the outcomes of in our research can be seen by all of you people, and you can say that the outcome of these searches up to the point the which our today's society requires that thinking. So I'm saying again on this platform that all of you are welcome who can collaborate with us on any part of the research with, under our strength and your strength. So with these words, I again thank organizers for inviting me to be a part of this important summit and share my views. I hope the outcome of these summit will be very useful, not to the researchers, to the researcher, to the scientist, to all the students, and there will be a lot of new things which students can learn from the outcome of this seminar. Thanks again to the organizers who inviting me on this event. Thank you.
Thank you so much, sir, for your encouraging words. We are definitely looking forward for more collaborative research works. I shall now request Professor Dr. Ronindro N. Shaha, former Director, BITS Pilani, Dubai campus, to kindly address the gathering. Sir, please.
Good morning, respected Chief Guest, Dr. Ravichandran, Pro Vice Chancellor, Dr. Pandey, Dr. Shajal, Dean, Dr. Aluk, indicates I'm becoming older. My senior actually Aluk, but close friend also, Dr. Rajat, my close friend, from whom several years back, when he got connected to Adamas, I heard of Adamas. Because we are very close to each other, we used to talk whenever I was Dubai or Pilani, whenever I used to come to India, Kolkata, because it is my native place, I used to talk to Rajat always. So, and dear faculty members, dear students, BITS Pilani has a system called surprise test. What is the surprise test? You go to the class to teach. Students do not know. Then while teaching, you can start 15 minutes before you stop, then write the answer, or you can have a paper with question, give it to the students, they have to write the answer. We used to call it surprise test. And it's a part of the curriculum and part of the course, offering like full end of semester test, open book test, similarly surprise test. So today, Adamas also has taken my surprise test. Because it was not part of my a request what I received from Dr. Nandan and Dr. Partha, they said only you have to deliver a lecture. But suddenly when I came and sat there waiting for lecture to start, because teachers have always a problem, they like teaching very much. And that the hours are up hours. That's why my wife always says, please don't start teaching to me. Okay. But anyhow, thank you for invitation. It is always an honor. I'll not take much time, don't worry, because I'll trouble you more afterward. But two things I'll tell you. Grab the opportunities. I just saw that Ayush Calcutta Center scientist has delivered a lecture. He suggested collaboration. Grab it. When he says academic industry, it need not mean physically meaning of industry. It can be a R&D organization. Ayush, I know, they offer many application-oriented projects. During COVID, they came out successfully, some oral anti-COVID, I don't know success, etc., but they came out. Many such things are there. I request the faculty members, that is the meaning, purpose of doing this conference. It cannot be only discussions and forget next day. I'll request administration, Pro Vice Chancellor is there to push the department. Go and visit Ayush, have a many meetings, and see how a project can be obtained. I'll request faculty members to do that. Then real success of this conference will be there. I have a request to students. Please utilize that. In my lecture, I will discuss that. If real collaboration happens, students get the first benefit. I'll tell how. But you don't get the benefit, keep in mind the materialistic benefit. Oh, I'll get a 5 lakhs, 1 crore annum job. Your target should not be that. Keep in mind, if you expect 1 crore annum salary job, you must earn 5 crores to 10 crores for the company, because company will not only pay the salary, because they have a cost of building, light, and so many, then shareholders' profit. That means if you earn 5 to 10 crores rupees you can earn, then only company will give you 1 crore job. So your target should be to prepare yourself that can I deliver things to the company? That means your target should not be the job. If you prepare yourself very well, that yes, any company I go, they will rather interested to take me because I can contribute to the growth of the company. Then rest of the things automatic. Because you have a good knowledge, every company will be interested to have you because they know you can deliver them very well. Then rest of the things will be automatically. Same thing I used to tell my faculty members, I have only request to you. Please, we should not target, I'll be professor after 10 years or 5 years or something. Rather, your target should be, I should be a very good teacher, I should be a very good researcher and contributing to the university. If you publish very good quality papers in the reputed journals, you teach it very well, what is required for the promotion? These are the things required. So if you do these things, automatically your promotion, you have to expect your university will give you the promotion. So your target should not be. Then what happens, oh, okay, where I can publish something? Okay, can I contact with my friends and join, get a publication? Then your research is not developed. My point is only that this discussion in this conference, I will request the administration, department, faculty members and students to take it forward. Why I am telling students, if your father in some industry, it need not be only pharmacy, it can be biotechnology, it can be engineering side. Connect your father to the university and see how collaboration can happen. That is the way BITS Pilani grew, where the new admission students, their parents are very high position. We had a separate meeting with the parents. That, okay, can you come and have some collaboration, irrespective whether it's a pharmacy or not, any engineering or anything. I think that should be the approach of the university authority. That should be the approach for the department. One last statement I will tell. I normally don't remember names, but people told. If you want to walk for a mile, you have to start with a single step. Okay? So start that step today. I'm sure you will reach your goal. Once upon a time, 70s period, I used to sit like you, same thing, Dr. Ravichandran, Dr. Rajat, Dr. Alok, we used to sit like you as a students, where people spoke. If we can come to this side, you can also come. Keep in mind. And you should have a target, but not, oh, I should go by manipulating, requesting, by provide. No, your achievement, you come and sit on this stage. If you come back after 25 years, where your university inviting you and asking you to sit there, can you imagine what your psychological achievement? Keep that in mind. Nothing, money, etc. Rest of the things automatically will come if you be successful. Thank you very much for taking it.
Thank you so much, sir, for the enlightening words. It's very nicely said that if you have to walk miles, you have to start with a single step. Definitely, I believe that our students have encouraged themselves from the wonderful words. I'm waiting for the students to grab the opportunity and learn something from your lecture. Now, it's indeed an immense pleasure to have amongst us Professor Dr. V. Ravichandran, Director, NIPER, Kolkata, as our chief guest. Thank you, sir, for your presence, sir. I would request you to kindly address the gathering. Sir, please.
To all of you, the management, the Vice Chancellor and the Pro Vice Chancellor, Jitendra Pandey, invited the guest, fellow colleagues, Professor Shaga, Ghosh, Dr. Sarkar, Rajat Ray, Sajal Shah, and our invited participants over here, the faculty and staff members. I am really delighted and happy to be part of this at the today event. Healthcare is that the important objective. I was hearing from that you were organizing the team. So really it is a complexity. Healthcare, it is not so the simple component to address with one simple logic with a few points on that, the healthcare. Is how the complexity going to be solved. Last couple of years, from 2020 to 22, almost we are nearing to 3 years period, how the pandemic the taught a lesson. The science is very important for that, the survival of the human being, the healthcare. Our country is at performing the best possible level, maintaining the health in the country and extending the same technology to the other country, that level we are very strong, no doubt on this. But whether it is sufficient to address the requirement, it's not so. There is a need of some additional requirements, the scientific data and the technology, what it is need for matching the global requirement, matching the health aspect, it is very, very important. Though initially we are trying to address that the crisis, then how we are going to attend the medical needs, unmet needs. What are the unmet needs now that the patient centric? The problems in the health sector, it is not that homogeneous. It is a patient centric, it is a very heterogeneous. So how the preparation of that the young generation to address in this aspect of patient centric, the technological aspect as well as the health aspect. So logically, how we are going to prepare our next generation in the knowledge based and efficiency based. The biologicals are very important. The biological in the sense, you all know that the communicable diseases is one aspect, non-communicable disease, chronic problems, non-communicable disease in the cancer and the chronic problems are that very huge task for the human survival. You know that the biological means immediately it comes to our knowledge, the antibodies. If you are taking the treatment for any problem for 1 week to 2 weeks period, the antibody, if you have one single dose, you can correct the problem for that from your emergency. Then it's a maintenance dose. It is there. That's the way that the vaccine we have started and we are surviving. We scared about the problem to come out and to assemble together. Now we are all sitting here. The biologicals are that a very important logic over here. But still there are more technology it is need. Where the focus it is need is that very much in this area. Antibodies, antibody drug conjugate, where the technology is very minimum in our country. We are importing most of the technology over here. All the gene synthesis is it is imported from other country. We don't have that any major component, though we have that gene synthesis smaller segment we have, but we don't have that any major aspect of the KD level. It is a major level, antibody level. It is not there. We need to focus on this area very much. And what role we can take? End-to-end research, translation, there is nowhere in that the country it is established. Still some of that the central university system, AIMS, IIT, CSIR system, ICMR, they are working together to strengthen this aspect. Still there are some gap on that. The segment wise we need to prepare ourselves, we prepare the scholars to enrich in that the technological requirement. The segment wise in the sense that the designing of the work, what is the current status, how to design that the material, how the material can be converted as a product, what are the regulatory requirements for to attend that the safety and efficacy of the material. Being a pharmaceutical people, the kinetics is the very important component. How that the kinetics knowledge will improve that efficacy of the material. Now everyone talks about that, the site of action, very specific, the targets, to come down, to bring down that several of off-target problems
or the toxicological problems, toxicity problems. This is always major challenge. You all should think about that. How specificity, that's the reason now everyone is pushing into that bi-specific material of antibody drug conjugate. One that the component it works in that the one target, material carries that into the target. The specificity it works well, efficacy is improved, toxicity comes down. This is a mantra and logic of that, how to handle the kinetics for that, keeping the product the best level. You all should understand that. That is a biologicals, it's going to rule that the country. The biological in the sense it is not that the simple or single component. There are several segments starting from that vaccine, antibody. There is a RNA intervention, DNA transcription reverse technology on this. Logically, how we are going to learn certain component here. This is very important. If you understand that, then you will be part of that the leadership, as Professor Saha conveyed that very rightly. So one day you are sitting here, you should come here with that 10 wings of Ma Durga with all the techniques and technology. You sit here, we all again come and enjoy that your the strength and the technology, how you are very efficient on this. We will be very delighted and happy and proud with this. This is a very important component. What are the challenges in the futuristic challenges? Geriatric population going to be double during 2050. The people are acquainted with the quality service, acquainted with that the people, what you want, that high quality, I need that the high quality of that the service. Everyone it is becoming old, and geriatric population going to be double that the strength of the percentage with the 2050. While you are in that the service, see, you should focus on that the personalized medicine and patient centric area, where it is these all the component can extend that the quality service to get that the major job on this. These are a very important component. And other than that, you all know everyone talks about AI, artificial intelligence and robotic, the technology. The artificial intelligence and robotic technology, how it is going to be helpful. What robotic technology, it is why that artificial intelligence and robotic technology it is going to be a useful one. The reason is that to bring down the wastage, the man-made error it is that the brings more wastage component. It could be possible to have that robotic technology and AI-based technology can bring down that the wastage. It is very important. The corporate, always the company what they expect. So what you can give, there is a demand and supply. The employee what you can contribute for that the industry. This is what the industry is expecting, investor is expecting. As a teacher, how you are going to contribute to that the profession for the training of young leaders. Why this the way that the university system, the department get that the higher credential. The credits to the teachers, it goes more and more by that the scholars. It is a compliment for that the system, and it is a compliment for that the scholars also. So what we are going to do, this is a very, very unique and important component. The technology now that it moves from that the multiple dose containers into that the single dose containers and disposable components. Earlier what that the normally eye drops and then the vials injection, they use that the multiple dose, containers. The system is completely now it changing into the technology of the single dose use and that the throw of that the container. It even in the eye drops. Once it is open, use and the throw it, that the container it is thrown out. Otherwise, that is so many complications it is faced in that the formulation container. And again, the batch of that the manufacturing process, the larger batch of that 5 lakhs to 10 lakhs unit of the dose, it has come down into that the 10,000 to 15,000 level, the dosing. The minimized simple dose level for which how that we are going to make ourselves that efficient to handle that the minimum batch size from that major batch size of that the million level. These are the very important component. We should prepare the young team members, the next generation people to understand that the smaller batch. What is the big thing in the smaller batch? It goes to the level of personalized medicine. The clinician, once that the prescription comes over there, then the dispensing, it starts with that the smaller batch of that the 10 dose to 20 dose level. So how it will be that the patient-centric, the preparation can be done. The personalized medicine can be done. It starts from that the material of that the flow. It's a flow technology. It's currently ruling the pharmaceutical. I don't know that exactly now the current the challenge in the flow technology. It is very very minimum. Flow technology. It is I can say that there is no flow technology. Only in that the R&D level it is going. But it is a need of the knowledge, need of that expertise, resources in all aspects, either from that institutional level or from that the industry level also. The flow technology, it is the raw material aspect. The raw material, it goes to that the formulation aspect also. How it will be a useful measure for that improving that the yield and bringing down that the impurity level, and cost management also involved in this. The batch size into that, the flow technology, it will be a major logic on that. We have to improve on this. I believe the colleague has come, so we are very happy that he is always the part of activity in everywhere. The manufacturing process, how the documentation is very important. The documentation in the sense, you may think about that the document of the preparing GLP requirement and GMP requirement. Now these all modified regulatory requirement, modified that the document in the aspect of online documentation. All the data in the quality control for ensuring that quality assurance, online documentation, the manufacturing, even it is a manufacturing process or it is a quality control testing, the kinetics data, all it is online monitored system. The document is that very much ensured for empowering that the quality of that the material. It is that the quality assurance goes in the level of that the online documentation. It is forever, you cannot intervene in that the document data. By this the way we should prepare our people on this documentation. So online documentations are very very important. So you can go for that paperless. There is no paper, the registering and then again modifying it. The raw data can be modified later stage. Such kind of that the system is completely winded up and modified into as per that the requirement. What you are doing in Adamas, it would be monitored by US FDA by online. If you connected that, they will see that what you are doing, whether that the reagent loaded in right time, sample is tested in periodically with 6 hours, and what is the data sounds, they will see that the entire monitoring process sitting there in that the regulatory division. So the regulatory division modified the requirement in such a manner. You have to think about that how you are going to address in this aspect, preparing yourself. The imaging it taken that the major role Imaging, it starts from that, the photograph. What photograph? It goes into that, the maximum in the omics level. The omics level, what kind of the transcription it happens, translational aspect, what changes it happens. These all the component image, it's very important for any of the data to ensure that the quality molecular medicine level imaging is improved in such a level where you have to focus, you have to learn the things, and that the testers what you could able to do, you have to ensure and understand that, make it as powerful skill, one of the skill for that, the imaging, it is a molecular level requirement. Medical imaging, this is based on that artificial intelligence, also part of the component for ensuring the data. At the personalized level, the clinicians are more want of that, the highly skilled pharmaceutical person for the handling of chronic problems by extending personalized medicine. I don't know the Partha what he is going to do that. I have given certain presentation with him. Yeah. So I believe that it is these are the component where basically the complexity of healthcare need to be addressed segment wise. So I am really very excited to be part of this event. I wish to encourage and enrich that the science, the research component. You can address to the teachers. So we are really excited. We have that around 30 number of the testers we have listed in our NIPER. Based on that, what facility we have and expertise we have. Among these, the 30 testers, where you are fixing into that, which one you want to learn for your employability, your technology transfer, or that you want to make that the entire the society, the healthcare system by simple technological aspect, whatever the concept you are bringing, you can make it very well and bring it wherever that the strength is there. We are here to encourage you. What even I can say that whoever getting the best prize and best award today, we can the sponsor that the project, if it is comes in that the realizable manner, in collaboration with NIPER, I ensure that the project will be executed in collaboration with our institute, our facility, and our people will mentor you that in such a aspect. Thank you so much. Yeah, with this, I end here. I believe they have that the break, tea break. Then we can continue that. I am happy to be the part of this event. Thank you for the organizing team.
Thank you so much, sir. Now it is the time where we are going to release our e-souvenir book in the conference. I would request our Honorable Pro Vice Chancellor, sir, Guest of Honor, Chief Guest, and our Dean, sir, to kindly do the same. I would request the IT and the media team to assist them regarding in this matter.
Thank you, sir. And thank you to all the delegates. We are now at the end of this inaugural function. To give a perfect end to this session, may I now request our convener, Dr. Nandan Sarkar, Assistant Professor, School of Medical Sciences, Adamas University, to deliver the words of gratitude. Over to you, Dr. Nandan Sarkar.
Hello. Good morning, one and all present here. A very warm welcome to all of you in the 3rd National Conference on Healthcare Industry and Academia Summit 2022 and beyond, organized by School of Medical Sciences, Adamas University, Kolkata, in collaboration with Central Ayurveda Research Institute, Ministry of Ayush, Government of India. This conference will provide an ideal platform to the participants' interaction, creation, and dissemination of ideas among the distinguished healthcare professionals, eminent scientists, technologists, and industrialists. On behalf of School of Medical Sciences, I am glad to be standing here to express my vote of thanks. Firstly, I would like to thank our Honorable Chancellor, respected Professor Dr. Shomit Ray sir, for his precious time out and making to this event happen. Secondly, I would also want to thank to our Honorable Vice Chancellor, Professor Dr. Naveen Das sir, and Professor Pandey sir for time-to-time help and guidance. Also, I would like to express my gratitude towards our respected Dean, Professor Dr. Sajal Kumar Jha sir, and our HOD, Professor Dr. Sanjoy De sir, for the event in order to make it a success. Before going to the eminent voices, I also feel extremely happy to thank all the faculty members of School of Medical Sciences, organizing committee members, staff members, non-academic people, and the people behind the stage, especially the media person, ID people, for their continuous support and complete preparation for this program in short span of time. And of course, I would also like to thank a heartfelt thanks to the student volunteers members for their participation. In this such a short of time, you all have made this best arrangement for this program. Thank you wholeheartedly. On behalf of the entire institute, I would also like to thank to our eminent guest of honor, Professor V. Ramachandran sir, Director, NIPER, Kolkata, Professor Dr. Ranendra N. Shah sir, former Director, BITS Pilani, Dubai campus, and Dr. Nishant Chakraborty, Associate Professor, School of Medical Sciences and Technology, IIT Kharagpur, Professor Alok Shah sir, for accepting our invitation and for being with us today. Your thoughts have truly inspired us, sir. We have received an overwhelming responses from almost all of the part of the India of the participant and would like to thank everyone for displaying such great enthusiasm. As a convener, I extend my warm welcome to all our participants. I am very much confident that the participant will be able to upgrade their knowledge and skills pertaining to healthcare through interactive sessions, including deliveries, talks from these our eminent speakers, and oral and poster presentation from the researchers showcasing their research. Finally, I would also like to thank each and everyone for being present here today and making this day a wonderful one. Thank you.
Thank you. Thank you, Dr. Nandan Sarkar. It is time for us to take a short tea break. We shall return sharply after 15 minutes, around 12:25, for our first technical session. Thank you. I would request the volunteers to kindly help our guest for the tea.
আছে না নেই? হ্যাঁ, লাইভ আছে না লাইভ নেই? নেই তো?
ensuring that the cost also in this aspect. So the regulatory requirements guidelines are nowadays very stringent and very important. You should understand that. Before you select that any question, any hypothesis, concept, you should understand what are all the requirement for that, the regulatory requirement to attend that the translational research or that the application based. At the present stage, what the burden healthcare cost is? It's huge. There is no medicine, there is no technology for the COVID, but the charge used to come that 15 lakhs to 20 lakhs rupees. This is the tragedy. You all should understand that how the business is going. There is no rationalism. There is no identified drug, specific drug. Only that by choice. The diagnosis is also very minimum component. Now it has improved into that. But the charge going in the business, how it is going? More than 2 million level. You have to understand very carefully why science should work on that to solve this kind of the problems. It is a very, very important aspect. In the pandemic, already I conveyed that how the government working and worked on that, extending the several million, and it has crossed billion also, exported more than 75 countries. This vaccine is exported to the 75 countries. That level we are strong, but still it is not sufficient to attend that the complete requirement. If any the futuristic aspect it comes into that. Now the problem is coming to that the patient centric, not that the overall it is a component, it is a common aspect, it's a patient centric logic. It is very much important. How we are going to be very specific. This is the very, very significancy in that the scientific and technological innovation. Scientific technological innovation, where currently we are in the generic, we understand that this is the raw material, this is the formulation, this is the quality control, the regulatory requirement. In this aspect, our business is going, but we are in that one small segment only. We could not able to take more business aspect in the global market. This is the major problem we are facing. You are being that the learner for that future generation, you should focus on that scientific innovation, technological support and the scientific support and technological innovation which can improve on this. In the future, where it is the future is with biologicals. The future is with the biologicals. It starts from the normal diagnostic to that, the treatment, antibody drug conjugate, several therapeutical material. Theranostics are with the biologicals only. Biologicals, biosimilars, where if you understand that in the sense of molecular level, you should understand how that the DNA transcription, reverse of the DNA transcription, how it works, RNA intervention therapy, oligonucleotide, nucleic acid therapeutics are the very important component to resolve that the problem of major, the cancer, including that the virus, and it extend into that the bacterial problems also on this. These are the very important area we should understand. Though the diabetes it is a major, it can be controllable. Diabetes induced the problems. Diabetes induced neuropathic, nephropathic or cognition are the major issue in that the diabetic. We should understand that diabetes is not the problem. Diabetes induced secondary issues are major problem. There is a focus, there is a need, it need to be attended. By this way, cognition improvement, neuropathic pain and neuropathic disorders, and again there are several liver failure and kidney failure, mainly that the nephrotoxicity and that the kidney failure issues are major and major aspect need to be focused. what is the market logic here? That non-communicable problems, it's the huge market. Though the communicable diseases is the market infectious problem, it is there. Non-communicable diseases, it is the potential is more and more the chronic, the medical problems. Chronic diseases are very huge. It need to be attended very rationally. Technological advancement, what happens in the technological advancement? It is a imaging, it is a one of the important aspect using artificial intelligence in the molecular medicine. It gives that together in that the theranostic and go for that the rational prescriptions. These are the very important component. The focus with that the drug development, the investment in the drug discovery. As I conveyed earlier, again I am repeating, we are good in that the generic, not in the discovery. There is a need of that more and more discovery part, diagnostic part, and again that we should use the component of personalized medicine. All that the cases are not same. The secondary problems complicating that the primary issues where to be focused very much. This is the personalized aspect, it is very important. The focus of personalized medicine as well as that where the innovative technology, innovative companies are, it is acquainted by that the current industries are that the major. You can be a startup, you can be a entrepreneur after 10 years period with a technology. Now that the industry looking here the best technologist as their employee. You don't think that with the qualification of that the B pharmacy, M pharmacy and that PhD you will get a job. If you go with a technique, the technique what I conveyed that either in the kinetics aspect or in the molecular medicine or in the diagnostic aspect or nowadays it is going to be the major task in this bioengineering area. The bioengineering area is that very important area for that even material science, for material productivity and then bio-ink area. These are that the very important for the doing of 3D printing. We are hearing so many information, technique and technology and the product on the 3D printing. What is the reality on this? Whether the material is available, material is not that easy for that making of that the 3D printing of any product, for which the bio ink is that very unique. You should understand that the physiology and the pathological condition accordingly, that the tissue engineering, bioengineering of these materials are very, very important. It is called as an bio ink. It is a very basic component. If you enrich the knowledge in this area, you will be the wonderful person needed for the company. If you go with the techniques and technology and the skill, the company will welcome you for a job. And as that Professor Saha was saying that if you go with that 1 crore, if you want 1 crore, you should give 10 crore. Then only you will get 10%. It is not that you will get 1 crore, with that 50 lakhs your contribution. The contribution should be tenfold, then you will get that one-fold. It is ensured. The consistency of the same thing should be ensured. It is not that one-time magic also. The regulatory, as earlier conveyed the same thing, regulatory requirements are very stringent. Regulatory people are that working for the society as well as that improving that the business in that the society. So it is a balanced way. They should work for the government or regulatory segment, work for the government as well as for the industry people. It is not that only one person. I can say that if you are looking your mother, you are not looking your that the wife, then you will be in trouble. So you should be very, very careful. You see that mother is also important. The balanced way, wife is also important. You cannot ignore that in one area and concentrate in other area. Then you will not be respected in the society for any aspect. This is very, very important in our culture. So it is a similar kind of logic of regulatory is that for that the best society, because government is committed for the society. And the same manner, government is committed for that improving that the industry also. So there is a balanced requirement. That's what it is a navigation regulatory hurdles bringing that the product to the market. It is not by urgency. You give that to say, oh, I am submitting so many detail. You keep on asking that the detail. Not that they keep on asking in the sense. It is not only the efficacy is important. The toxicity and safety also is very important. By this the way the regulatory requirements are very much it is needed. You all know that all that the components are there, all that the components of the data are documented online data. Whatever testing, the testing is that the online testing, it is controlled by the regulatory people, and whoever that collaborator or partner, they are looking into that what process it is going by online. You should understand that how that the online data are that coming into picture. It is not only with that that certain technique. How to connect with this the component of current regulatory requirements are very important. Whatever it is, at the end, how that the cost of the product, the price control is vested with that the government. They are looking into that, give that the quality medicine with affordable cost. This is a very important the mantra of that the government policy. Late to market, what is that the efficiency and emphasize on that the maintaining the confidentiality. These are very, very important on ethics. Because a company developing, investing so much money, and the data confidentiality, people working there, managing the personnel, that within that one group of that 4 or 5 the team members, within a family of that 4, 5 or 6 family members, so many complexity it comes. So in this aspect of managing that and maintaining the confidentiality, you should have ethics. Someone investing money, someone paying salary for you, you should have rationality, ethics on that, then the growth will be higher. For both the aspect, it would be the complementary, either investor or a company or employee or a team leader, whoever it is. If you are a damaging person, within that short period, you will be knocked out, like editing the gene and putting in that somewhere dustbin instead of for that application aspect. So while we go for that the traditional medicine, what are the bottlenecks on the traditional medicine? I am going to give that certain the data, how it is useful in our sample, or that the traditional medicine means India is the number 1. Business is for that the China. Why? Why business is for China? It is all that the components are India, but business is for the China. The reason is that they are keen on that attending the global regulatory requirement. We are saying whatever language I talk, that is correct. You should not ask that any data. This is for the several 100 years we are using the same material. It is correcting, it is useful. I know that my grandfather used it, my father used it, I am using it. You should accept it. No one is ready to accept it unless otherwise you give that scientific data, technological innovation. These are all the components. It's copied from India to China. They are making money. They are keeping all their people very safe in this aspect. So only this is the traditional medicine in China and India. India is that it's a mother for that the aspect, but we are losing our royalty with the China. But their economy is the best way. We are losing our ground in that the base by phase wise. What are the problem in that? The quality control is the problem on this. They will give that the material, they have written that 1 to 3 spoon. You are all the technical people, including me, that we should solve the problem of that, how significantly this dose is optimized. This is the dose with that the 5 milligram. If they are saying it is that the traditionally with that the 5 gram means, how you can bring down with that kinetic knowledge into 5 milligram. That is your technical skill on this. It's a very important aspect to ensure that the 5 milligram available in each dose, and confirm that this is available end dose also with after that the 15th dose also. It is very important to ensure that the quality control aspect. Now that the cellular target, what I conveyed, there are several segments of that. It is the target binding, cellular target, without binding with the any off target, then the efficacy, toxicity is less, efficacy is that very appropriate. The cellular target is very important. Mechanism of action, how that the expression it happens in that the omics level, like transcriptomic level or proteomics level, whatever it is that reverse the DNA transcription level, you have to ensure that the component of that, the mechanism, clinical validation on this aspect, how many the patients it is utilized, the data sounds on that, the clinical validation, complexity of the herbal formulation, the complexity of the herbal formulation, how that the complexity, now that without that 2-3 combination, there is no material comes. No material comes. But at the same time, there is need to be ensured. I don't say that the Ayurvedic or Ayush products are not good. But what the scientific knowledge, you are that the technical expert or your skill on that going to ensure that. Few of the component we can make it very clear. The very clear in the sense, we should ensure that the dose, we should ensure that the consistency of the dose, we should ensure that the testing of that the component. So all this the data support for that more than one herbal formulation. One herbal formulation, phytopharmaceutical, it is that the new procedure for that, the phytopharmaceutical as a new drug. But in Ayush component, Ayush medicine, more than one herb, it is that the utility in regular practice for management of several diseases, it is practically it is there. How we are going to address the issue, how we are going to maintain that the consistency is the major challenge. If you understand this, the language, skill and the technique, then you will be that the rich person in our society itself, need not go to any other country, need not go to that any other company, you can make your own single product on this. Then that would be a great, it's a business for you or earning for you that. Logic here, any herbal product. There are omics nowadays, whatever the scientific contribution, scientific evidences are, very much it is available. That's what it is a need for that, the scientific community or regulatory requirement to frame that regulation on this. What omics, any material, it is subjected in the biological system, it extends that expression of that the protein, the transcription it happens. The transcription starts from that epigenetic modification, gene expression to that proteomics expression. These are all the data sounds on that. Then you can see that these are that the expression data it is available. It ensure that the scientific component ensure that it brings down that the abnormality of that the protein which causes the problem, pathogenicity. It brings down that. It intervene with that the RNA intervention, it happens and modify the protein expression. These are all the component you have to ensure with the formulation complexity, combined action of that, the different target would be the complementary on this. In little bit elaborated manner, how that omics going to be happens here in that the DNA level, RNA level and the protein level. So whatever the components it is listed here, all to be, you can understand that. How that the proteomics, RNA therapeutics or that DNA modification or in the aspect of metabolites, metabolomics components, all together if you understand in one segment, if you understand this omics, this is a simple picture. It looks simple. It looks very simple picture. It is not that easy to understand that the complexity. The complexity is that very huge. You have to understand each segment wise. If you understand this each segment means what I am trying to say. If you understand that RNA intervention or that DNA transcription, or if you understand that how the reverse Robotech is that now the new technology, it comes into that RNA intervention technology. This is that small molecule which destroy that the RNA of that pathogen, or that even in the cancer, Robotech technology, it is available in the current the technology. It is going to change that entire therapeutics. That's what I conveyed. Biological going in this the way where we are learning. Really, we are learning something. It is very important. What the technology movement it is there, what the industry is looking into that. These all the components are very important. You cannot blame that any teacher. You cannot blame that any institute. You should understand, update that where we can focus. Whether we understand that the 100 technique. If you understand that one technique among that the 100, you will be that unique and wonderful person. Still elaborate manner. How this is omics comes from that the genomics, cytomics, proteomics, metabolomics and transcriptomics. These all comes that in bigger beings level as therapeutical requirement. I will give some space time also here. You can understand that it is not that that simple component, but you should be very clear learning of this the component to ensure that your employability, then that the industry is looking this the way. If you know that only the definition of that what is the transcriptome, what is the proteome, proteomics aspect, RNA intervention, then they will give only definition equality only. So you should understand how we can get trained in the proteomics, transcriptomics and RNA intervention. I can say that a practical component, remdesivir. The people were fighting, throwing stone and that nonsense. You are not giving remdesivir for my mother is dying. Why? What is the reason? Reason is that RNA intervention technology which controls that the virus. The same thing, same RNA intervention technology controls that the cancer. These are nucleic acid therapeutics where that the gene modification is the problem and gene deficit is the problem. That is a rare disease. If that the gene deficit is there, it is a rare disease. Certain functional aspects are lost. It becomes the rare disease. Certain gene modification happens. Epigenetic modification it happens. Mutation it comes to that. Methylation, demethylation, acetylation, chromatin modification, histone modification. These all together makes that the complexity of that. The mutation, there the transcription it happens, then naturally the DNA functional aspects is lost. Then what is going to happen? So DNA functional aspect is lost with methylation. There is no aspect of the functional, then it continue into that the several problem of complexity of disease progression end with the cancer. So where we are going to learn, where we are going to improve our skill. This is what it is a challenge. Really we should think about that. So we want that, no, I have that additional hand more than that existing two hand, additional hand, how that this is a skill. So you should prove that what additional hand you have. In the application aspect, how that the genomics going to be a useful one? The people in the Ayurvedic Ayush product, they are looking into that. There is no evidence. But the China made all this evidence, it is all in there in the scientific logic. In the global market, even in the US market, these products are sold there, they are using these in the US market. But there is no rationality. We are failing to convince our own family people, our own people in India. We need not convince that anybody else. We need to convince our own family and our colleagues. That is the society. If you have that knowledge, you have the skill on that, you should understand, start from that, this is the basic component. I have all the evidences for this material. This is the material A. Everyone knows that haldi is that works very well. Where it works and what it does and where is that the data on that, the curcumin, whether it is several the million works it is done, why that curcumin it is not come as a drug, it is not listed in the monograph. What is the reason on that? There is no clarity on this. For basically we need that these kind of that specificity, certain modification and that omics data, it is very important to ensure that the logically, even with that somewhere we may fail, but this scientific component, if you learn this scientific test, then your job is to ensure your recognition is ensured. I am very much confident on this. So to understand that even a small molecule also, now that the logic of several components, it comes as a target. But the epigenetic modification, it is very important for that prevention of the disease. Not that symptom-based component, symptom-based one single protein, it works with that epigenetic modification. It is basically, if it is reverse at the DNA transcription aspect, then really the mutation is stopped in the basic level, the problem will come down. Otherwise, that the symptomatic, only superficial, like that sugar management, if you attend that the glucose level management, there is that the productivity or that any the insulin deficiency or any other factors influencing that the controlling of resistance of insulin. So all this component segment to be taken care into that, identifying the material. Again, I want to say that the people identify diabetes, we are working on diabetes. No one is expecting it the diabetic, more than 100 rupees, what you can do that diabetes. The 100 rupees is available with you, 1 month you can manage the diabetes. The people is surviving for that more than 40 years with only that the metformin and the other available simple material. 40 years the people are surviving. Complications of the diabetes are that very huge. The task we should understand. Selecting the problem is that major important thing, that while you go for that any employability, any company for interview, you should say that this is the question I have selected because this is a high task on that. So I don't want to stick on here that with the genomics, transcriptome, proteomics, metabolomics and lipidomics, cytomics. These all that the component need to be learned and understand that for your higher validity. We are going to see that one case study on that a material, how that the Chinese medicine, integration of transcriptome, whatever we have shown that we have seen last previous couple of slides, transcriptome, proteome, metabolomics in the system pharmacology. That to reveal that the therapeutic mechanism underlying Chinese herbal Bifidobacterium formula, it is going to be listed as a BYF for the chronic obstructive pulmonary disease COPD. What they have done on this logically by using that the omics. This is a model for that. Any component if you are selecting, if you ensure that the transcriptome and that the proteomics component and metabolomics, it will be a ensured data for that for approval or that acceptance with the scientific community. This is mainly used for COPD. This is the material BYF. What that determine the mechanism, how that the COPD for that integrating transcriptome, proteome and metabolomics with the system pharmacology data sets. At the present stage, what is that currently the people talk about that single target protein. Reality of that single target protein in the symptoms, it is not so it's correct. Why it is not so correct? Any pathological problem, it is associated with the multiple problems, multiple proteins defect. One protein it is modified, any antigen enter into the system, protein-protein interaction, protein modification, it is not that only one protein it is modified. There are several proteins are interacted and modified, it loses its functionality. At the end, we are getting of that this protein, because of this protein losing its functionality, this is that the symptom, it comes as the pathogenicity we need to address. That's fine. But down level, down regulation and up regulation component, whatever it is that the different proteins, it is upregulated, few protein it is down regulated, what are all the different pathway analysis associated, cell signaling, together it comes into that the picture of modifying the transcriptome and proteome. It is together with that the metabolome, what it is that modified, any material it is consumed, directly this material not going to work with the target. The material get metabolized, segment wise it gets separated, and metabolome it binds with that the different proteins, and it ensure that the functionality, it create that the action. So by this way in the product, whatever it is the crude material it is consumed, subjected, it modified, metabolized by that several enzymatic changes, and this material again, material in the sense pharmacophore bind with that the protein, it ensure that the activity. Logically, the COPD, you know that the problem associated here in that the 3 omics data with the gene, proteins and metabolites were predominantly associated with that oxidoreductase activity and antioxidant activity, the focal adhesion of that lipid metabolism. This is what it is the exact the segment here. We should understand that the transcriptome, proteome and metabolomic data set. What exactly the detail associated here? Inflammatory response, oxidative stress and that focal adhesion. These are the 3 segmented role on this. This is the analytics of that the potential targets of this formulation in that regulated gene, protein and metabolites. It shows that exactly how, what are all the different changes it happens. What is the conclusion here? We are coming to that end. Beneficial effect with that COPD. What is the beneficial effect? The COPD is controlled. Like several material in that the Ayurveda, it controls that the problems. But systematically, we should have the transcriptome, proteome, metabolome. The logic you should understand, not only the component of the wording, you should have that scientific data and innovation on this, then it will be supportive for that. Here, inflammatory response, oxidative stress and cells functional pathway in the system level, it is modified and it's corrected. Still there are several aspects it is there. The want of time, I am not going into that, the level of how that it is the intervention of that, the pathway signaling, it happens, JAK-STAT pathway intervention, and again that protein interaction, these are that the complexity it is associated. What are the disease focus in the institute currently it is going in the NIPER? Neuropathy, inflammatory bowel disease, non-alcoholic steatohepatitis, it's a NASH, Alzheimer, and the renal dysfunction. These are the identified area we are working. If you have that skill, if you have that some testing, the technique, you can collaborate with us. I extend welcome for that, invitation for you. If you want to collaborate, yes, I have this thing. I am doing it. It will be complementary. What you are doing more than this, that fivefold, I can do it. I am very happy to give you. No need any person to say that I have that committee there. They can assess you that what is your plan. This will be at a one stroke it will be done if your plan is that very efficient. Okay? This is very important. I am not here only that give something talk, give something at the, get some bouquet, oh, wonderful coffee. It's very good biscuit. We all cross that, Professor Saga and Ratatreya, everyone that we cross these all, saying we cannot take anything because of that, the people reception we are taking, this is more than that what we need. It is not at all correct, but still, it's a reception aspect we are using and we are respecting the recognition and acknowledgement. Why I am saying here, these young people are very important for future generation. The country should not beg for technology in that after 20 years. Begging technology means what? What do you want to say? Whether we are begging, the reality is that. That's the reality. Because the remdesivir, it was not available. We were facing so many problems and crises. The people called, and why you were a director, nonsense fellow? You could not able to give that remdesivir a few injection. That what is the meaning of you are a director? So it is a uneasy thing, but availability, non-availability, and affordability also is a problem. What the reason is that it doesn't mean we don't have that any scientific strength on this. We have enough scientific strength. We are not understanding that reality. Once it comes to your family, comes to you, you understand that reality of that, the problem of nucleic acid therapeutics. By realizing that, I am telling you that the nucleic acid therapeutics is not a simple thing, that it is currently what money we are spending for this purpose of managing the cancer, managing the virus. Such a level of huge problem is there. We should focus very clearly. Even I can say that for this also nucleic acid therapeutics, I am not talking the simple component. We have 10 crores project. I am very happy to train that any of the faculty or that interested PhD scholar in the area of nucleotides and nucleoside synthesis. We have that the collaboration aspect of the NIPER with the NCL and again with cultivation science. We are together working on that couple of the Morpholino. We are designing this material, we are synthesizing, and we are trying to go for that antibody drug conjugate of that Morpholino also. So the point I am trying to say that you should come out from that your comfortable zone. You cannot come here by that the 10 o'clock, leave by 5 o'clock, something we have done that 3-4 classes, we have taken that 2-3 practical, my job is over, we will sit there in the canteen and finish it, that this will continue. Okay. I believe that I am not going to finish within my time. I am serious about that completing my time. At the same time, I want that you should be tracked in that the right direction. While we are here together, that we want that drive you, monitor you in that the better the direction. You all know that what the progression it starts, what the progression of the disease it starts, whatever you eat, whatever you talk, whatever environment you are there. So together it create a epigenetic modification. This situation makes into that the disease condition. Physiology into that, the pathological condition, epigenetic modification, it happens whenever that the demethylation of DNA it has stopped. Your problem of disease is started. Pathogenicity it started. You should understand that mutation, it is that the started, it continues into that inflammation setting the tone for that the cancer. This is what it is, that it leads into that fibrosis and the ending with that the cancer. This is what it is, the inflammation which involves that activation of recruitment and action of that cell innate and adaptive immunity. While we go for that the pathogens, inflammation is equally important for that repairing of the cells. The inflammation, it is not only that it making the problem, inflammation also it is very important for that repairing of the cell, and the inflammation, it is a basic for that the developing into a cancer also. Neuropathy aspect, what the neuropathy here, the diabetes induced neuropathy, nephropathy, cognition, these are the major secondary problem associated with the diabetes. So this is the association here, it is that peripheral nerves and that the damage in the fibers, autonomic nervous system, all the damage happens. This is a very major issue after 20 plus year, after 2 decades of the diabetes, you can realize that the problem of that in the geriatric cases, neuropathic, diabetic neuropathy. The mitochondrial dysfunction, we may understand that what the mitochondrial is. The mitochondria, it's a power center, that is one aspect, it's fine that. How that mitochondrial changes, it comes into that. How the mitochondria going to be a target on this, it protects that the pharmacological modulation of AMPK and SIRT1, this is the important components over here. How that the diabetic induced neuropathy with the rat system, it is a work is under process. That targeting inflammasome in neuropathic. Inflammasome, you understand that this is a segment of the inflammatory component in the brain, how it works with that, the leads into that, the damage of that, the neuropathic neurons, damage in the neurons is all the major issue associated. These are the data with that it is given as Bay11 7082. NLRP3 is that inhibition aspect. These are the data associated here. And how that the next problem is, next portion is the inflammatory bowel disease. Even in the inflammatory bowel disease, we are trying to develop the organoid. So why that the organoid and what is the requirement of this organoid? The people started to do some component in the cell line, animals. The reality of that the problem is vested with the individual. Individual, that is a personalized medicine. The individual patient to patient, it is at heterogenic. The problem of it is based on that how secondary problems are associated here. Either it is a sympathetic or asympathetic associated, and the problem of the disease IBD, inflammatory bowel disease, it is all based on the practice. How that we are ensuring that our olden culture of that, the food practice, as well as that the environment, what the work environment also it is involved, and the practice associated here. Why IBD? It is that because of it, the huge problem, it starts from that, the IBD to that several organs. I can say lipoprotein, you all know that lipoprotein. Lipoprotein with that the butyrate, it works very well. Butyrate, it is a small chain fatty acid. It is an important component of that, the gut microbiome. You can see that the butyrate small chain fatty acid, how it is very important for that the health. Health means including that the cognition, mind power, mental power on this. The people, they go that several places, my child is not performing well. The problem what they are feeding, how they are maintaining that the health gut component, and how that butyrate it works well. This is a very important component. So what other people can give that some mantras or the sutras for that managing that the mental power. This the butyrate is very important for the cognition. Butyrate is important for that, the several aspect of that, the apolipoproteins and lipoprotein conversion with alpha secretase, beta secretase, gamma secretase, beta secretase and gamma secretase, which promote that lipoprotein into beta amyloid, and then it is a promoting factor from the people always think about Alzheimer means it is in the brain. Where it goes, it moves from that the gut only through that the promoting factors, beta secretase and gamma secretase, the liver, lung and the heart, everywhere the beta secretase, beta amyloids are settled, and it makes that the problems for that the health system. So why we have selected IBD? IBD, it is in US, and then next to the US, Asia and India, this is a major problem, and the problem, it is pathogenicity starts from here only. That is the main reason that to control over the IBD, it makes that the human manpower quality would be better, improve that manpower quality. This is that what the validation on that the NASH, it is one of the component we are working on that pathogenicity, how it develops from that the basic normal liver into that hepatocellular carcinoma. Different stages it is associated with simple macrovascular steatosis, fibrosis, cirrhosis, hepatocellular carcinoma. These are the pathogenicity, progression of the disease. Vasakudichi Kvata, it is one of the product by the Ayush. If you understand that, this is a product several hundred years it is used. It has that its own effectivity. The reason I am saying that this is a Vasakudichi Kvata, it has that several significance. Several significance means what? I was talking with you about the DNA transcription reverse. It is not a simple component, but this material, it reverses that the DNA transcription. By this way, it is very important. While some material intervention in the transcription, it is not so simple material, but the cost of the material is very much affordable, and there is no major side effect on that. What you all people should do here, the consistency of that the dose and what the material it is present need to be ensured. The quantification and consistency is very important in each and every dose. Then it makes that very sense with this the product which useful for that the simple management of the problems. What is the work plan? These all that the collection, other things, so you all very much aware that, but the plan is that very important to get into that this product. What are all the segment wise material it is present by that the mass wise, whatever the small molecule it is present, it has been listed here. So what are all the small molecule? You could see that the small molecules, it's all well known small molecules, whatever it is available. It is with all the material, it is a common material. You could see here, luteolin, I believe that one of the faculty also working on that. And again that funicalin, fumaric and cebulic acid, corilagen, this is the corilagen, cebulic acid, together it is there in that the terminalia material. These all that the material it is present, but what you have to think about that, these the component secondary metabolite means in the natural product pharmacognosy we will read, we could read that all this, yes, these are the constituent it is present. It is not only in the aspect of that the only constituent it is present, how this the component can be fixed with that or bind with that specific protein, how you can enrich that the pharmacophore efficacy into that higher level and specificity are very important over here. While we go for that quantification, it has been very carefully quantified with that corilagen, berberin, gallic acid, luteolin and vesicinone, cebulic acid, cebulinic acid, ellagic acid, these all that the component, how it works binds with that the different proteins. The omic is that very important logic here. So how this pharmacophore bind with this all different proteins. This is a network protein associated in the during the pathogenicity, either the upregulation or the downregulation aspect. These all the components are that identified 9 compounds with that binding with 32 targets, which makes that the controlling of this problem of the NASH. Animal model of the NASH, it is all the initially it's starting with that what specificity of that the product, then how it is at the fatty liver, NASH, and then fibrosis, hepatocellular carcinoma level, how it is improve. The problem goes into that from steatosis to that the NASH. Animal model it is. These all the component, it is a diabetics model associated NASH, it has been tested, this is the data for that. What are the different material used for that, the controlling of biochemical analytical data sounds about that, significant decrease in the level of that glucose, AST, ALP, ALT, triglyceride and the cholesterol level. Again, subsequent the data for that, the product, what it is in the different concentration, dose level, it is used here. This is the significance of that, the histopathological study data. Well, this is the transcriptome data of that the product. In continuation of the functional differential expression of the genes, proteomics analysis of the same component, how that the transcriptome it is done, the similar aspect of the proteome it is carried out. Proteomics analytics here, what are all the material small molecule and how that, what are the different proteins it is at the expressed, upregulated, downregulated. In the row 1 it is a control, the row 2 it is at whatever the rate component, you could see here that it is a diseased pathological condition, then how it is modified into that the different segment, the imaging sounds very clearly. I conveyed with you that imaging role on that artificial intelligence, how it is very, very important on this, that the omics data, it is one of the important logic, the same component you could see from here with this, what it is we could understand here. Control and the disease model, how this that the disease model modified into that with after subjecting this material, what are the modification? These all the blue lines are that modified the expression of the protein comes down under the control. These all the red line in that second DC, you could see that it is a diseased control, the normal and diseased control. From that the NC, the normal into that, after subjecting that the dose level, low dose, high dose, or this level, you could see that all the you know, the imaging of that the blue color, it ensure that it is proteins are recovered into normal status. Again, it is the continuation of all the component. We will come to the conclusion. What that the collectively, it is effective with that NASH. The NASH is the major problem. NASH is the major problem mainly with women. Women after the age of that the 50 or 60, little bit bulky, they may not go for walk or that any exercise. They sit ideal, it become the NASH. The individual become that the NASH. So the liver problem starts there. Liver problem end with that the cancer, liver cancer. Further clinical study would validate that the findings, it is a need. I end here. I thanks the organizers. I'm sorry, I believe I have taken some additional time, but I could not able to control my time, but I believe something it is taken 15-20 minutes additional. So any questions are there, you can ask me, you can write to me. I am very well responsible for that. Any data we have given, I am very happy to. So get your input on this also in all aspect. With this I end. Thank you. Any question is there before I go there? That I am not saying that I am going. I want to.
Thank you so much, sir. Now I would request Dr. Partho Roy to kindly give the token of appreciation to our speaker. Sir, please.
Just on behalf of School of Medical Sciences, I would like to thank Professor Ravichandran for that encouraging speech. And at the personal level, on behalf of all the faculty members of School of Medical Sciences, it's our privilege that Sir has invited us to do collaborative research. So definitely our faculty members will take up this opportunity and connect with Kolkata NIPER as well as NIPER Hajipur to do some collaborative research. And next time we are on stage with Sir, I am definitely, Sir will show us some slide of collaborative research with Adamas University. Thank you, Sir, on behalf of Adamas University.
Thank you so much, sir. We will now move for lunch and we shall meet again. Okay. Yeah. We will be meeting again at 2:10 here in the auditorium for our next technical session. Just one announcement. The students, Adamas students, will be having lunch at the room number 308, 309 and 3010. I'm repeating once again, the students from Adamas will be having the lunch in room number 308, 309 and 3010. Sir, it is in the pharmacy building. So, any volunteer, please help in taking him to the pharmacy department. The scientific committee over there will be helping in finding the room. So we are going to start our session now. I would request all the participants, students and faculty members who are present over here to please take up the seats. So welcome back one and all for this post-lunch session. Our speaker for this session is Dr. Ronindo N. Shaha, Senior Professor at Department of Pharmacy, BITS Pilani, and he has served over 35 plus years in academics. During his tenure, he has held many academic as well as administrative positions, including Dean, Deputy Director of Administration of Pilani Campus, Director of Research and Educational Development of BITS Pilani, driving research and related agendas for all 4 campuses. He also served International Campus of BITS Pilani at Dubai as Director for nearly 8 years. Just excuse me please. Sorry for the inconvenience. He also served International Campus of BITS Pilani at Dubai as Director for nearly 8 years. Before retirement, he also served as Vice Chancellor Acting of BITS Pilani, consisting of 5 campuses. He has executed many projects sponsored by industries and government funding agencies. In 2013, he has received Pharmacy Professional of the Year award given by Indian Association of Pharmaceutical Scientists and Technologists for his contributions in pharmacy field. He is also the recipient of the Best Pharmacy Teacher Award for the year 2005, awarded by Association of Pharmaceutical Teachers of India in recognition of his contribution in teaching and research in the field of pharmacy. Apart from these, he has also received numerous national and international awards for his teaching and research. He has published more than 125 papers in reputed international and national journals, and more than 100 papers have been presented in international and national conferences, seminars, and symposiums. He is on advisory board and selection committee member of number of universities in India and abroad. He is also member scientific associations in societies like APTI, IPA, Controlled Release Society USA, Association of Pharmaceutical Scientists USA, American Chemical Society. Presently, he is serving as visiting professor and consultant to some pharma industries. It is indeed a pleasure to have you, sir, amongst us. Thank you, sir, for your precious time. I now request you to kindly take over the dais for your presentations.
What is that here? Oh. What is that? Cordless speaker I need if it is there. I give you pen. Lapel is better. লোড করে সেটি দেখছি কোথায় ফাইলটা that boy? Pen drive is also not with me.
So it is there.
Okay. Sir, later. Which one?
This one, sir.
No. On, yes. Now it's okay? Yeah, okay, fine. Anyhow, actually, without that also I at least I have experienced that. Okay, good afternoon. Till I get my pen drive, which I gave it to some volunteer, because I find it is not loaded here. Anyhow, thanks to the organizers. I will not take much time on the introduction, greetings, etc. But you know, the speakers for the sessions before lunch and session after lunch is big challenge. You know, before lunch, everyone is in mind, I have to go for lunch, otherwise it will be crowd, I will not get my food properly. So having concentration is a very tough job. And post lunch, all audience will be feeling drowsy. So I have a big challenge to keep you all awake. Anyhow, I'll try that. Please. So I'll try to hold so that you don't sleep. Particularly the last bench, they will lie down below the front, back. So I'll try to hold it. Anyhow, what I'm going to speak about the theme that industry-academia relationship. We discussed, I remember when I was student also I heard these things, how it is a very important. But I don't think that much of the things have been done at all places. Yes, few places it has been done, but it always needs to be taken care more. So what I'm going to speak is industry-academia relationship, scopes, benefits, with some real information case studies and ways forward. And keep in mind, this is important for university administration, faculty, department, as well as students. You have also a role to play because it is your benefit, keep in mind, that this industry-academia relationship is important for students. Why I am telling you, many times we heard in the industry when we go, oh, today's graduates are not industry usable. That means when you graduate, you are not readily useful for the industry. That means they feel that there is a gap between the what you have learned, what is needed in the industry. And that's why many companies have started now, they conduct finishing school. They use the term, oh, we are conducting finishing school or finishing program. That means they take the graduate, give them something so that they are useful for the industry. But we do not agree. I personally do not agree of that, because as a university we have the responsibility to make the graduates useful for the industry immediately on graduation or before graduation. Why university provides the education? For economic development of the students and their family. Because if I prepare you very well, a well graduate, you will get a good job, you will earn money. Okay, what is, I am going to speak. My content, but don't worry, I don't have 50 slides. I have only 11 slides apart from my title slide and thank you slide. You don't have to bother and go on counting how many slides are there. I think about 11 slides. What I am going to do, what is industry-academia relationship? What are the different facets of industry-academia relationship? Multiple facets, some of us probably even don't know that one. I will be also discussing some case study, some relationship which is in work successfully, which is all working for decades. Benefits of this relationship for department, for university, for faculty, for students. Way forward, Is it just I'm going to sell theoretically, oh, this is important, how to do it, how to implement it? I have some ideas which I can try to put it, and obviously question and answer session if you get a chance, if I get a chance. As I mentioned, it is a very old theme, always discussed it, but how much is achieved? Not much, as I tell you further, but keep in mind, this relationship has to be win-win for both sides. It cannot be only benefit for the academics or university, it has to be benefit for the industry also. Only then industry will participate actively and will contribute in the academic program. So that's why when we design such system, we have to keep in mind how industry is going to benefit from that. I'll try to discuss that also. If you see what is the relationship, I am trying to say that, if the relationship, internship of the students, what does it mean? Just not spending 1 month. It has to be built in the program. It has to be part of the program with credits against the course. That means what? If you are going for 3 months or 6 months, as a part of the program you are going, it will earn a grade for you, then you will be serious to do it. Industry will be interested to get as much as possible from you, and faculty will be responsible as the industry personnel. Can you just control it, or I am speaking? Why this sound is coming? Okay. So all stakeholders will be there, faculty, for evaluation, so that a grade is awarded or marks is awarded to the student. Student will be serious to contribute there, so that he earns a good grade from that things and he will force to learn it, and industry has a stake that they will be taking as much as possible for you. You can say, what is the benefit for me? You have a multiple benefits. You get a real life problem from this internship. You get allowances, pocket allowance from the company if you contribute to the company. Is it a dream? No, it is not a dream. It is happening, I'll just share with you. So it is the internship of the student which needs to be part of the program with credit connected to that course. Obviously placement, though it is a separate, but I mentioned here because that is also industry academic relationship. What is the other facets of industry academia relationship? Offering degree programs to the industry personnel. Universities should think to offering some program, degree program to the university personnel. I'll say again, many universities are doing it, not all. That is the important part. There is a contribution can be made by the university. They have to properly think of that program and offer it to the industry personnel. Conducting training and workshop. Many areas. I myself conducted workshop in pharmacokinetics, what is it called, IVIVC, many other things. I conducted training program for the industry personnel. That is possible. 2 days, 7 days workshop for the R&D staff or other staff, which in university can always conducting the training workshop. That is another facet of relationship. Using industry personnel for teaching. I heard Dr. Alok Ghosh used to come here. I don't know how much you got it, but I know very well what in Ranbaxy, in Lupin, how much contribution he made it. He brings a real life problem to you, which I tell you as a faculty member in BITS Pilani, I could not contribute that unless I talk to the industry people regularly. So he can bring many knowledge to you, which will be useful for you when you graduate and go to the industry. More such people should be invited to the academic institutes, conducting separate course, part of a course, or if required full course. Many times it is possible he can come, an industry person can come and stay 7 days and finish extensively part of a half or 1/4 of the course. Faculty members, you have to think of it. You decide, in my course 25% of the content will be covered by industry personnel. Connect to them, invite them, they will be happy to do it. I tell you, it doesn't require much cost also. That is possible using industry personnel for teaching. Industry immersion scheme for the faculty. Faculty should think of spending 2 months, 3 months or summer vacation in industry. See what are the things going on there, what type of projects they are doing it. They can participate there, see the problem or discuss with the industry what problem you are facing, which problem you are unable to solve it. Learn that problem thoroughly 2 to 3 months, come back to the university and submit a proposal, work on that, submit a proposal. I am ready to solve your problem. Are you ready to sponsor that project? I think this is, I am giving a name called faculty immersion scheme at the industry or industry immersion scheme for the faculty members. That's another relationship. Sponsored research projects is a very important part, I tell you. I'll show how it can be successful. So sponsored research project, I'll be discussing more details on that one, where you can get, if you have ability, you can get the sponsored research for the industries, which can be done at your university and develop the technology and deliver to them. What is the benefit? You get fund for the department, you get equipment from the fund, you can get many other things. Then your students will be working on that project, so they work on a live project which is important to the industry. Instead of doing a project which is a more theoretical or more of just my idea, but if I have a project from industry, you are getting a better exposure to the industrial project which is going on. Even a BPharm student can contribute part of that. It's a group of students can work, MPharm, BPharm can work together and it can be successful. I will show you some successful stories of that. So sponsored research projects is another one. TLC or TIC, Technology Innovation Center, it is opposite to sponsored research. It is a industry will come to the university to get some work done. You may think, what is that? It is possible. Sometimes a smaller pharmaceutical company may not have a good R&D facilities. They can come to your facilities if you have a good facilities. They can come to your department with the problem. He can visit, supervise along with the faculty member the project, so the result output can be taken back to the company. I tell you, today when I was traveling, I have a message from a company. I am reading the message. I always try to be very connected to the reality. When I was in the car along with Dr. Roy and Dr. Ghosh, I got a message from a company, Calcutta-based company having good R&D. I am not giving the name. Sir, during your visit to Adamas, please check if they have a particle size analyzer. We can give small projects to them. This is a real message. I can show it to you. I talked to the faculty members, I talked to Dr. Roy, do you have the facilities? So that means if the facility is there, this company is writing me, sir, we are ready to give a small project. Start from a small project, morning what I said. If you have to go for a mile, first step is always need to be taken, which is probably 1.5 feet. If you do it successful, they will come in with more project. Then it can be a big project, because one successful project can bring 10 projects to you. Again, with my experience, I am telling you. So I can share the message with the dean, but if you come forward, I can connect to the company. So this is where the possibility that industry may come with the project and get it done at your place if the facilities is not there. A company R&D person will be coming 2 days to you so that he can get the sample analyzed and take the result there. Extending R&D facilities to each other, this is another thing. If the collaboration is there, industry can come and use your facilities, or you can go to the industry and use the facilities. This is very possible. It is not impossible. Many in university or industry having such type of relationship. You can think of that relationship. That is something, another facet of industry-university relationship. Establish industry-sponsored named research laboratory. Many universities, including BITS, we have many companies, Microsoft, SD Microelectronics, they created a laboratory with their staff deputed there, where the students of the university are allowed to do work, whatever it is. Their requirement. There is an agreement should be there. If something comes out of who will own the technology, all these things are there. But I tell you, Hyderabad campus, Goa campus, Pilani campus, even my Dubai campus, we created that. And we have named laboratory which is specifically sponsored by the company and where work is done related to that company. Establishing the industrial sponsored named research laboratory. Establishing business incubation center for the startups. I think each university should think of it. For that is required an innovation center. I tell you, when I moved to Dubai in 1914, I had a dream that I wanted to create an innovation lab or innovation center. What is this? I established within 2.5 years, I established a laboratory which is not attached to any department. I clearly told it will not be owned by any department. It will be 24/7 open, because I have a hostel in the campus. I said any student midnight suddenly some idea is coming, he can enter to the lab. It is a biosensor based entry. He has to put his finger. He can enter into the lab, whatever idea is there, he has work. I have provided many facilities there. They can work and it will be owned by them. Believe me, within a 1.5 year, 7 students team got sponsored project from the UAE government. They introduced a student project, which is a 50,000 dirham. 50,000 dirham means multiplied by 20, 10 lakhs rupees, Indian rupees. 7 students group got with that whatever they have developed, they submitted the project. Then they said, government said that now you have to implement it, give a model or whatever, working model, etc. so that it can have a business possibility. And many of the students with their developed project, they have made a startup company, because before I left in 2021, when I moved to Pilani as a Vice Chancellor, I ensured that one TBI, Business Incubation Center, created in Dubai campus, 4th in a row, because earlier Indian campuses had already, so that the students' company can work in that Business Incubation Center. I'll request the faculty and dean, you can think of a similar, but it should not be given to any department. Then, and no faculty can control. I clearly guided that no faculty control the project. No, this project is not possible. Because as a teacher, we have a problem sometimes. We feel whatever the student's idea is there, that is bakwas. Please, madam, sirs, I am telling you, come out of that. You don't know today's generation what they can do, which we feel it is a absolutely bakwas, can be a very, very important thing. Allow them to do whatever it is. You have seen sometimes a small kid making something, we get surprised. I mean, how we can make it. So give them that freedom. 24/7 it should be open, no control, nothing, but yes, responsibility is there, no one should spoil that one. That's why biometric system is there for entry. And it is running very well, I got it. I have the photograph, I did not want to take many of there. One more thing I mentioned, missed probably, extending doctoral opportunities to industry personnel. In the university should think of that one. That means industry personnel can do PhD without dislocating themselves to university. No required for them to come. I shared today, West Bengal got a new governor, am I correct? What is his name? Dr. C.V. Anand Bose. He is not a medical person. Do you know his doctor DR has come from BITS Pilani? He did PhD because I was that time in charge of PhD program. He is an IAS officer in Kerala that time. He joined BITS Pilani PhD program under that university industry collaboration. You can say IAS, how can be industry? That's why I said industry is a common term, but I say it's an organization. He was that time director of Kerala Nirmiti Nirman Kendra, which is working on a low cost housing scheme. He joined that time BITS Pilani PhD program, and it is not easy. Like any other students, whatever required, BITS Pilani has a PhD qualifying examination. Those who know American system, you must be knowing it, that American system candidate has to appear a PhD qualifying examination. BITS Pilani has a qualifying examination consisting of 3 written papers followed by a viva. Believe me, Dr. C.V. Anand Ghosh fully came to the campus for 5 days. 2 days he took time to prepare, etc., final preparation, then appeared in the 3 days written test of each one 3 hours. But you will be surprised to know that is open book. But don't think open book means you open the book and write it everything. No. Open book is the most tough examination. And BITS Pilani system, minimum 30% should be open book. I can come and conduct workshop on open book examination. So he appeared for the interview 4 days. Then he went out. After few days he was announced, not because he is IAS officer, because he did very well. He cleared the PhD. Then I cleared the PhD qualifying. Then he started his PhD work, got supervisor, but he need not require to come to campus, stay 3 years there. He did it work there in Kerala, ultimately submitted the thesis, which went to the examiners, he cleared it. I can proudly say that Dr. C.V. Anand Bose, who is the new governor of West Bengal, is PhD from BITS Pilani under that scheme. 1989 he started. I was fortunately that time in charge of PhD program. But do you think it has been opening a floodgate to the industry people from earning money? No, sorry. Because PhD program doesn't allow any earning, frankly speaking. Because examiners fee, getting examiners for PhD, viva, extensive expenditure. Till 32 years, I think not even 100 people have been allowed to admit under that. It is a very selective. I'll show some more cases also, even pharmacy cases, but that is another opportunity where university can do a havoc. I don't know how many of you thought that these many opportunities can be there for industry-university linkages. I think of that creating the innovation lab, I'll request to think of that one. Allow the students to do whatever they like. but keep a monitoring, but not controlling. I'm taking a case study of BITS Pilani Pharmacy School, but many of the things are for all programs. For your information, BITS Pilani, for every program, this university industry linkages is there. But I'm giving the examples of pharmacy program. Right side is the internship. Practice school, which is 7, and you can't believe probably, out of 4 years program, 7.5 months they spend in industry in 2 parts. After second year, before third year, that is summer of between second and third year, they spend 8 weeks in industry, do a project which is called Practice School 1, which is a 5-unit course. So a faculty goes along with the students all over India, wherever the PS1 stations are there. They monitor the students' progress, and the evaluation is not study, etc. Whatever they see in the project in the industry, there will be group discussion, there will be quiz and many other projects, etc. Then they earn a grade, which goes to NCGPA. Then there is a 5.5 months duration for Practice School 2. That is half of your final year. Half of the students go in the first semester, other half doing the elective courses. Then this batch goes in the second semester, and those students come back to the campus for doing the courses. And some of the companies, name my pharmaceutical companies, I gave the name: Lupin, DRL, Dr. Reddy's Laboratory, Torrent Pharmaceuticals, Ipca Lab, Sun Pharma, CDRI Lucknow, Om Laboratories in USA. Their students was taken from BITS Pilani. They paid the airfare, they paid the visa, they paid their pocket money to stay in the USA. And they 5.5 months they work in Home Laboratories in New Jersey, which is actually that time is a Ranbaxy's company. It is in abroad also is possible. I'm not saying everyone at this moment running, but it is overall whatever. Novartis. I remember I was instrumental for Novartis in 2002 or 3 sometime. I was visiting there, they invited to deliver lecture, they said we were the practice school student. For each student that time, for M.Pharm student it was 35,000 per month, and for B.Pharm student it was 25,000 per month in 2003-4. But they used to take for pharmacovigilance division, they used to take the students that one. So these are the some of the companies I mentioned. Deloitte, you can see probably Deloitte is there. Deloitte is also there, probably I have not mentioned there. You can say Deloitte, how can be? Deloitte have a vertical in pharmacovigilance, they take the pharmacy student. TCS have a vertical in healthcare, they take the pharmacy student, biotech students. So that opportunity is there. PhD, we have students from Ipca Lab, Ranbaxy, Torrent, Colorcon, which is a excipient in the company, Strides at Collab. We are offering degree programs for Dr. Reddy's Lab, Lupin, Sun Pharma, etc. For their manpower, we are offering 2 years MTech degree in pharmaceutical operations. So their B.Pharm students join in that MTech program, and it is they are working, they are not coming to my campus. They are working in the company, they are also studying for the program. Classes are held in the evening or Saturday, Sunday, and which can be technology through video, through distance mode, or even my local faculty can go and teach there. So Hyderabad campus faculty can go to the company and deliver lecture. Otherwise it happened, Lupin I know, they have a, what is it called, HR division in that near Aurangabad. What is place? This Bombay and Pune between? What is this place? Very tourist place. Dr. Alok, your HR, Lonavala. So my faculty used to fly on Friday, Saturday, Sunday, Monday they used to teach the staff of the Lupin, and Tuesday morning they are coming back campus and taking the campus courses. That is possible. We run that way. Then we have a workshop conducted for Torrent, Sun Pharma, Lupin. We conducted workshop pharmacokinetics, IV, IVC, and the other biopharmaceuticals and other area. Sponsor project. We have got sponsor project from Ipca Lab, Torrent, Dr. Reddy's Lab, Colorcon Incorporation, Lubrizol, US, Colorcon US, Imami, Geol Life Sciences, BASF, etc. I tell you, BASF have a scheme where they provided free excipients as much as possible to the department. So for your research work, any excipients of BASF you need, you should have a collaboration and get all these things done. But since something you do for them also. I'll show some of the success stories also. Yes, etc. I told you that where you can see the map of BITS Pilani Practice School Centers. It is all engineering, not pharmacy only I am telling. All subjects you can see. All over India you have the practice school stations or internship program which I am telling you. Pharmacy I listed many names. Internship which I mentioned there already. Lupin Lab, DRL, Torrent, GSK, Novartis, Ranbaxy, Panacea, OM, many more, non-core area, IQVA, Pharma S, Kineps, Deloitte, and fellowship is 15,000 to 50,000. I got the recent information, not 30,000, 50,000 per month. Now morning what I say, if they give you 50,000 per month, you understand, you should be able to deliver them at least 1 lakh to 2 lakh rupees business in that way. But you contribute, I'm not saying you will be getting the sale, etc. No, you contribute, which has a value of that. Then they will be ready to pay you 50,000 rupees. That means you have to prepare yourself to contribute on that. So total practice school session, if I say engineering already in India, 250 plus organizations are there. In 7 years in Dubai, I created a company about 100 plus for the students of Dubai campus only there. 100 companies up there. PwC, PricewaterhouseCoopers. They take our students for practice school, pay big amount of money and then recruit them. The benefit of such practice school too is that if you contribute well, they will recruit you because already they know you have done very well. They will offer you, and companies pay 6.5 lakhs per annum, which is very good for pharmacy graduates, I'll tell you. MPharm students also can do dissertation instead of, they can do practice school instead of dissertation. So many MPharm students opt for the practice school, we don't object that, so that they can get their job after doing the practice school there. That is where the possibility. I'm not saying each and everything you have to do, but you have to work out something. This is, I was telling you, the work integrated learning program. You can see the circled one. We are offering a BSc program, Pharmaceutical Sciences, to the staffs of Dr. Reddy's lab. They have a lot of 10 plus 2 students, staff, for whom the career growth is stopped. So they came to us, can you design a 3-year BSc program? We did not offer BSc. On campus, we never offered BSc program. We have an integrated 4 years MSc. But for the industry people who offer this BSc program in pharmaceutical sciences, they are providing us every year 50 to 60 staff of them, for whom we are running the program. They are working as well as their students. Classes are held in that way so that work is not suffered, students also get the degree. Similarly, you can see another circled one, pharmaceutical operations and management. This is designed specially for the Sun Pharma and Lupin and few other companies. I mentioned at this moment it is offered for this organization. I mentioned, but I know some other companies also we have offered. Ipca Lab, we offered it. This is the opportunity. Some of you are thinking, oh, how it is possible. No, it is possible. This is a big list of industries for whom BITS Pillar is offering various programs, including MBA program, which is company specific. In electronics and many other areas, I am not wasting time on that one. You can see data sciences, engineering for specific company. BEL, Wipro Technologies. Wipro every year provides about 5,000 students to us for some degree program. I tell you, 4 campuses together, it is about 16,000 students, and our WIPD program, which is a virtual campus, we have 18,000 plus students sponsored by the industries. I will not take time. Industry sponsored research project is, these are the companies I am mentioning here, but I will say the outcome. You see this. These are the sponsored project and outcome of the products. Today you go to any pharmaceutical shop and ask for the product mentioned here, Movon CR. Movon CR, control release. CR means control release product. This is designed by BITS Pillar in my group, PyLab. It is patented and marketed by Ipca Laboratories. And we ensured that this part is written in the laboratory. Here is the written. So if you take the pack and see the label, you will find it written. We insisted that BITS Pillar name should be there. So this is made. Now patent means what? My students were all part of the patent. And then Ipca Lab commercialized it. Similarly, you can see here Zerodol. Another Zerodol CR. So these are the products which is patented then commercialized. But this is sponsored by the Ipca Lab, so I had to give the technology to them. And what is the benefits for the students and faculty who are worked with me? They got a financial benefit from that program. So my junior faculty got their first car from the project, first motorbike from the project. Students also got the money who worked in the project, because I told that time my Vice Chancellor that if I get the project, there should be a remuneration honorarium for the faculty and students involved. They said institute, university doesn't need any money. You take money but provide something to the department. So I created the facilities for the department, which is useful for the department. But my colleagues who have worked on that one, students who worked for them, I ensured they got the benefit also of that one. This is another product recently came out, tapentadol hydrochloride nasal spray. This is Torrent Research. We got the project sponsored, and you can see this is submitted, patent submitted by Torrent Pharmaceuticals in 2016. These are the people who are the owner of the inventor of the project. You can see my name, my PhD student, Bibu Jaya Abraham. He is a staff from industry. He did PhD under the project which is sponsored by Torrent. So she got her PhD. This is the company person also who are involved in the project, monitoring, etc. It is my name. This is my PhD student. So you see, from one sponsored project, two people got the PhD, one from the company, one from the full-time research scholar in the project. And the patent has been given our credit in the due recognition. That means that the inventors, our names are there. Then the product came to the market some time back. This nasal spray, this is a nanoparticulate liposomal preparation which have been got, you have to administer in the nose, and the directly drug will go to the brain. We have worked for another drug also, which I am not going to discuss, but this is the success possible if you are serious about it. So it is nothing imagination, and people got the benefit out of that also. I am coming to that. So what are the benefits? I am coming. They are mentioned why it should be done. This is a benefit for everyone. My faculty got, I got benefited, I tell you, but as morning I said, my target was not the benefit at the beginning. My target was to get some product done which can be useful and marketed. My whole dream was, I want to see at least one market product which is designed by me or my group. I got the benefit. But a spin-off benefit is the financial, because there is a royalty. I got the royalty from 2005 to 2010, and believe me, that time the royalty amount more than my annual salary at that period of time. But did I plan it for that? No, it has come as a just benefit of that one. It is possible for you. Think of it. If you can do, faculty and student, you will be happy to see that your design product in the market. How much satisfaction you get it. So how the process of implementation? I feel there should be an independent office, not by department around. It has to be university-wise, an independent office for the industry linkages with admin head, not run by a professor or someone. It has to be a full-time experienced person, a person who is having an industry experience. Probably that person can be very good for that one. And this head office, it is for the whole university, not only pharmacy. But contributions are required. There should be admin office with staff and budget. There should be separate verticals. Whatever verticals you decide. If you feel whatever I spoke is all bakwas, fine. At least you know that this is not the correct approach. I always used to tell my student that if you fail some research, it is a good learning for you. At least you know that this path is not the correct path. So if you feel it is all wrong, fine. Don't use it. But do something different then. But do something for that. That is the important part. You do something for that one. So, admin office with staff and budget, that is the responsibility of the university. That's why I thought that university dean of provision should be present there, if it is in the morning possible. Separate verticals. Decide how many verticals you want to clear. I am not saying all the verticals you have to start, but start with 1, 2 at least. Industry advisor required, like Dr. Alok Ghosh or some other people can be there as advisor to help that admin office. Faculty members should be contributing. They should be, some of them should be part of that one. Because a pharmacy faculty can bring the list of company who can be useful. There should be student volunteers, because main stakeholder is a student. So there should be some students in that administration process also, which will help by providing many support, or when the company people are coming, properly hosting them, taking care of them. That is an important. That which company we should try to gathering the information, providing the office. So that's why some student volunteers should be there. Technological support should be there. Advanced level technology should be given, not only a computer, a video conferencing, so that directly the head or someone can talk to the company people. No need of visiting there. So technological support should be given. That has to be the structure and the operation so that it can be successful. But you start with smaller part initially. finishing shortly. As I said, it's the 11 and 12 slide only, 11 slide. How to proceed? The first step, conduct a workshop engaging all stakeholders to work out the goals. You decide, you want to follow my things, whatever I presented there. But you first conduct, university to us, there should be a workshop. How do we proceed? Decide the plan, goal, how the structure to be decided, how much budget is required. So some decision to be taken on goal, how to work out goals. Structure creation, then you decide what should be the structure. Should be headed by an administrator or a professor. Can be possible. A professor can be, but he should not have any other responsibility apart from teaching and research. But he will have only that responsibility apart from teaching and research. I never say a faculty should be taken out of the teaching and research and only do administrative job. I did not do it. Even as a vice chancellor, I used to take a course. Clearly spelling out expected outcome. The team has to be clearly told that this is the expected outcome from you in a 3 years, in a 2 years, stepwise goal. Creation of advisory committee, identifying and creation of verticals, that which verticals we want to pursue, not necessarily all at a time, technological and administrative support providing, and involving faculty and students in the whole process so that it can be really successful. Without all these people, it cannot be successful, I can tell you. Faculty have to contribute there, students have to contribute there. What is the spin of benefits? Students experience real life problems and enhancement of their knowledge. I tell you, what you learn by working in the industry, in a 6-month course you cannot learn that. Please, if the faculty has that feeling, forget about it. I have another slide which I am not showing it here, that real face-to-face work is giving you more knowledge than a 50 minutes class. Students can earn while learn if prepared well for the internship. As I said, the big students are getting 15,000 to 50,000 students, 50,000 rupees per month from the company. So 6 months you work, you can understand how much you can get while you are a student and earning the degree. Upgradation of the program, introduction rule courses, because if the student goes, faculty goes to the industry, then he can bring the latest technology there and can think of a new course and addition to existing course. Because as a teacher, sometimes we teach only whatever bookish are there. But it can be a real life problem, a teacher can bring it and student can come back say, sir, I saw that new technology is used there. You get the knowledge. You can create a new course. Many of the courses are created by that process. Enhancement of the lab facilities. You learn, if you see those facilities today, what I said, you can create the facilities here in the industry, university, so that you get the project. So your enhancement of the lab facility will be there. Multiple benefits for the faculty, as I mentioned. Faculty get many things, including the financial benefit if they want it. University can be financially benefited by this process. Industry presence can enhance the academic environment in the campus. BIC can develop entrepreneurship and create new entrepreneur. I expect, please, many of you think of startup companies, new business. Why you have to work for others? I have a dream till now that I will have some startup and I am working that till. I have not lost my enthusiasm. I had a dream, but now I have retired, but I am thinking still I should have a company on my own. I am working on that. Not for profit only point of view, but entrepreneurship point of view. Enhancement of image and perception of the university. Because today's ranking, international ranking, national ranking, these are the very, very important perception of the people. So, and many other benefits is possible. But when you do industry, I always tell 3 things to be keep in mind for the faculty members I am telling you. If you work on a sponsored project, 3 things keep in mind. Deliverability. You must deliver to the company, otherwise company will not come next time. When first project I successfully transferred them, they give me 3 projects at a time to me. That is the benefit of that one. And it was not easy. I had to visit many times to the company to get my first project. But I successfully delivered the technology, which is a Movon CR, GDOL CR. Second is the deliverability. Second one is the confidentiality. You have to keep it confidently, because if you share with anyone else, other company may immediately work on that one. No company will like that his thinking is taken by some other company and brought the product earlier, which is very much in there. So you have to maintain the confidentiality. Third thing is punctuality. You have to complete it within the time, because if you delay it, company's necessity for the product may be lost by that time. Because they are thinking, I need to go to the market within 6 months, or 1 year, or 2 years. But if you delay to 4 years, by that time another company has launched the product in the market, you may not get anything there. So that's why punctuality is another important thing. So it is very important for university to have industry collaboration for overall growth and enhancing image and rankings. These are the campuses of BITS Pilani. I had opportunity to visit all the campuses, administer 2 campuses, Pilani as well as Dubai. This is BITS Pilani, I cannot leave that one. This is the temple is there in the Pilani campus, housing Saraswati, which is facing the building of the university, academic building, and blessings BITS Pilani. I always very much fond of this temple, which is any time you visit, you will see it's a fantastic temple. I'm finishing it here. If you have any question, if you don't have a question, that means you have not understood anything or you understood everything. So it is up to you to decide what you prove yourself. Thank you very much. So your teacher told you all that whenever speaker finishes, you must give a clap, whether you like it or don't like it. So you have given a fantastic clap. Thank you very much for the clap. Please be there, but there will be no question.
Any question from the students?
No question.
Sir, I have a question on
Oh,
behalf
sure.
of the students. So we have just started our M.Pharm courses, M.Pharm in pharmaceutics as well as M.Pharm in pharmacology. So on behalf of them, I'm asking how can they start with their dissertation work, as you said, in the company base. I feel that it is much more easy for the pharmaceutics to get a project in the industry based. But if we think of the pharmacology, how can they proceed with their work?
Okay. See, that is where the benefit having of industry collaboration. If you have a connection, you discuss with them what is the project going on. Okay, then they will tell you. So from that you decide. Instead of just theoretically, because you see many times it happened, I have worked on this in my PhD, so give it, whether it is useful or not. But the best benefit, I tell you, best way, without permission of the person I am telling you, you have a Dr. Alok Ghosh. He knows the real industry need today. So if you bring him and discuss possible projects, or he has a lot of friends, Dr. Roy, I know Roy has a lot of friends who are in industry and retired. They'll be very happy to come and spend time with you people, faculty members, and discuss that these are the presently going on problem on which industry needs a solution. You get the project. Give that instead of just because I have done the PhD on this area, so I should continue that. No.
Yes.
I think it should not be done. But if in the process you get a sponsor project, fantastic things. You put your M.Pharm student, PhD student, B.Pharm student, don't think, oh, B.Pharm cannot contribute. Please leave this concept, I tell you. A B.Pharm student also can do fantastic if you properly motivate and guide him. I have seen it many times. They have done better from the others also. That is. But be in touch. You know the real life problem. That is where you get the benefit of that one. Have contact more on them.
Thank you so much, sir.
I have a question.
Oh, sure. Tough question, I know.
No, it's not tough. It's very easy, a mundane one. You can say that the thing is that why the industry would be interested for us. You have given the sufficient length and breadth. You have given what is the need for the industry, what is the need for us, what is need for the faculty, for the students, what are the benefits, etc. etc. The other way around, to prepare ourselves, I need to know why an industry would come to us.
Why industry? Okay, sure.
Second question is that you have talked about the courses given to the industry. customized course for the industry. Now, its planning, as you have experienced, its planning, so universities has to go through the certain norms of maintaining the certain norms of for giving a course, like what you call it credit courses, the total length of. Now, is it possible all the time to fit in all those the requirements into the customer's choice? This is what.
Okay, organizer, can I take time? The organizer? Okay. Two questions he has. Why industry will come? So I'll give you the answer, but before I make a one-line statement first. Why industry came to me? No, industry did not come to me.
It came to BITS.
No, it did not come to BITS Pilani. Sorry, Dr. Roy. You don't know how many layers of my shoes I had to wear it out to get a project there. But I tell you the techniques I played. I started thinking industry project in 2000 onwards. Okay, fortunately I had some connection to the industries beforehand because of some different reason. I used to go to there, not a very big company, because of practice school was going on. That's another opportunity I had. So when I used to go there, I always present them. And initially it is my own money, but institute money. Or I have gone for another official work. I used to meet some company in Bombay. I used to go and tell them, I can do the project. Can you? Dr. Shah, very good. Thank you. I'll contact you shortly. But in 2-3 months, no contact, nothing. Again, I went there. Then I took one particular technique, I tell you. I told them that R&D person, I had a little good relationship. But no, in profession, no one bothers about good relationship. What I told them, that you see, doctor, you have 15 ideas of new products. But you cannot take all the 15 at a time because you have a limited space, limited manpower and limited resources, facilities, etc. You might be taking 5 or 6. Then can you give me one of the remaining 7? And you need not pay big amount. You will be surprised to know today, my first project cost was only 8 lakhs. But out of that, again I am telling you, about 4 lakhs I distributed to my students. I tell you how. I told whatever money you give me, you don't have to pay my salary because my institute is paying the salary. I may pay something, whatever that you have to bother. You give me money, but he provided all the drugs, excipients, it was a controlled release product. Okay? You provide all the excipients, everything, they transfer it to, they said, fine, I am ready to do it. Because if they do it R&D, Dr. Alok Ghosh knows, even an R&D scale is a big batch, am I not correct? But I started with only 500 capacity size of tablet, Dr. Roy. With 8 lakh rupees, where my expenditure was the basic chemicals for analysis and etc. Okay, and now I tell you the story, Dr. Roy. It was first product they gave me is a nimesulide control release because that time nimesulide was widely used there. I know. So they said, Dr. Shah, can you do it? We have a very good market of nimesulide. We have to go to a control release. We quickly developed in 1 year, developed the technology, just transferred the technology. That time, if you remember 2004, there was a hue and cry about nimesulide, that nimesulide causing a lot of, I don't know whether it is true or it is a simple commercial gimmick, I don't know. They said, Professor Shah, we cannot go now with the nimesulide. Can you do, we have a aceclofenac product in the market, which is a big market. Can you produce aceclofenac control release product, which is my mobile CR? Now I had a confidence because I have already created nimesulide control release product. I said, fine, I'll give you. Believe me, in 7 months I developed the project because already my nimesulide experience is there.
Did you do the scale up for them also?
My condition was up to R&D scale up. So my team, myself, I went there, sat there on their R&D factory, R&D facilities, and R&D batch we have done it. But although we told them, when commercialized batch you make it, my people will be there to ensure that scale up is happening that way. So my person went there to the China, Chennai or somewhere their manufacturing unit was there, Ipalab. So there, and we ensured the product was scaled up very well. Now that is where I said, remaining 7 which are unable to take it up, give me one of that. They happily did it because cost was 8 lakh. Now believe me, now today my project Colorcon and other Morningside is a UK project I am taking. It's a 40 lakhs project today. Today I don't accept anything below 25, 30, 40 lakhs project. But my expenditure is not at all for the drug and other things. My expenditure is basic. So what I do, I purchase the equipment, etc., and pay something of my people who are involved in the project. They are happily contributing that, and there is a competition who can contribute there. That is the way, Dr. Roy, I got it. It is not that they came, but now they come to me. Colorcon, they approached me. What is the Kalarkar project? I tell you, they have a lot of excipients. They market the excipient for a specific purpose. They came to me, I suggested them, I can work on a newer use of your excipients. If I prove newer use, that means their market widens. So Kalarkar project is going on. I have PhD students who are working in Pilani, who are working on some excipients, I cannot name them, and we are working on that, patenting of that along with the company also. We have got an additional use. If it can be proved, then Kalarkar will get a benefit out of that one. That is the reason they are coming. And what is another benefit? One Kalarkar staff is doing PhD under me, and a full-time PhD scholar also doing with me. This is a spin of benefits. I am getting 2 PhD students at a time under one sponsored project. But company person is not getting easily. He has to do the all grinding as it is happening that way. That is where Dr. Sai, it is possible, but as I told you, it requires an effort and tireless effort. You cannot get by sitting at home. First question. Second question you said? You see, that is what the BITS Pilani is advantage, but you have to take. BITS Pilani today is one of the first 6 universities sanctioned IOE, Institute of Eminence. You know, 3 IITs, 2 private universities, BITS Pilani and Manipal. Today we have a lot of freedom to do our own things, whatever you like. But don't worry, Professor Rai and faculty members, I am telling you. See, first you start with a short certificate course or training program type of things. Their technical staff may require some technical knowledge. Okay, you think of that. Today I tell you, you are auto university by your own rights. You can make your program. Why you bother about UGC, etc.? Degree, I tell you today, many others, should I give you a name? After saying BITS Pilani, many other universities started the similar thing today. You are university by your own. Start that, let them object. But if you are sitting with a fear, oh, UGC will object. No, you start, let them object, then you fight. You have to fight with them. Do you believe BITS Pilani is never part of AICTE? Till I was Vice Chancellor, they were pushing us, Shastra Buddhe, who is the chairman of AICTE. We are never part of the AICTE. We never go to AICTE. AICTE many times send a court letter or legal letter, etc. Why you are not following? We did not follow that. You have to fight that. Sorry, I know you are getting delayed. You have to go back. Any students? Sorry. IITs have a freedom. IITs can, you see, Dr. Roy, IITs can do. Why IITs not doing it? Now they are thinking of. Very limited way, very limited way. Sorry, I have taken more time. Not my intention. They have asked the question. Any student? I have my mail ID with the faculty member, Dr. Pasto, Dr. Nandan. If you want anything, please feel free to write to me. I always respond. It is never my practice. Miss any mail. If it is anything requesting, I will be definitely responding. You will get my response within 24 hours.
Thank you so much, sir. Thank you so much for this whole lecture. I know that each and everyone was highly energetic and listening, even after the lunch, everyone was highly energetic and they responded and understood each and every slide of yours. Thank you so much, sir.
I have noted some students left also during that time. While lecturing, I noted that one also. I am very generous person. You know? So don't invite me anymore.
No, sir, it was absolutely a pleasure. So, thank you, sir.
That comes to Megan. The dean is not here. I'll leave with the dean to invite both the friends again for a day-long workshop.
Yes, I was about to tell that he has mentioned about the workshop, and we will definitely plan for that, sir. It very much been.
But not only if I come for the workshop, first of all, you'll be worried, you will be getting a dangerous experience of that one. Because I try to be always tough, master. But if I come, I will be definitely meeting the student. If you don't allow me to meet the student, I am not accepting the invitation. So student, I am going to give you problem.
Thank you, sir. Sir, I would now request Dr. Rajat Roy to please come up on the stage.
Rajat is my 50 years friend.
So I would request Dr. Roy to please present a token of appreciation to our speaker, sir. Please.
This is for the staff.
Please give a big round of applause.
Today, best wishes for all of you.
Thank you so much, sir. I will hand over the mic to my colleague, Ms. Srilekha. She will be taking over for the next session. Srilekha, over to you.
Sir. Okay, so शुरू करो. Okay, so thank you, Joita ma'am. A very good afternoon to everyone present here. It gives me an immense pleasure to be among the most esteemed personalities who have won accolades in their fields of research. We shall now be resuming our session with the next keynote lecture by Dr. Nishant Chakravarti, who is an Associate Professor, School of Medical Science and Technology, IIT Kharagpur. Dr. Nishant Chakravarti leads the Regenerative Medicine Lab at School of Medical Science and Technology, IIT Kharagpur. Dr. Chakravarti is a physician scientist by training. He has done his MBBS and Masters in Medicine Science and Technology with a PhD from Queensland University of Technology, Australia. His research interest involves utilization of the concepts of cellular and molecular biology, biomaterials and tissue engineering to study human pathophysiology conditions. He has several international research publications, book chapters, and conference presentations to his credit. He is also an editorial board member of journals Molecular Biological Reports, PLOSON, and Frontiers in Medical Technology, and also serves as a reviewer for several international journals. In addition, Dr. Chakravarti is a national core committee member of Indian National Young Academy of Science and is an intellect member of Royal Society of Biology, UK. He is also the recipient of several awards, notably Outstanding Doctoral Thesis Award, Executive Dean's Recommendation Award for 2014, and Institute Silver Medal. Thank you so much, sir, for your valuable presence. With a huge round of applause, I would like to welcome you, sir, on the dais for your presentation.
Thank you very much, it's working?
I think so, sir. Let me check it once.
Yeah.
Hello, hello. Mute koda, sir. Hello.
Please connect. Yeah, hi. Thanks a lot for this wonderful introduction, and it did sound a bit too sci-fi for me. Anyways, firstly, thanks to the university and the conference for inviting me, and as I have been seeing since morning, it has been a fantastic arrangement that the convener and the team has done. So congratulations to the entire team for this lovely conference organization that you have done. Right. So now let's come to the talk that I have prepared for today's. And I would actually like to try this collar mic if it works. Okay. And do you have this one? Yeah. Yeah, I hope I'm audible. Fine? Huh? Yeah, okay. So, So since morning we have been hearing very fascinating talks, and I was really impressed by what Professor Ravichandran and Professor Shahad did talk about. So amazing talks discussing about the importance of how academia and industry can join hands together. And this is very important in today's perspective. Why? Because it's not possible only for the academia, which means the universities and the institutes by themselves, to deliver everything. Or for that matter, most of the things that we are using in our day-to-day lives, we see them coming from the industry. So it's not possible for them to work in isolation. So there has to be some point where they can join hands. And we are very fortunate in today's era that we see collaborations happening at different levels. Maybe at the level of individuals in the form of researchers working together, or in the form of institutes and academia working together. Maybe this is making some interference, sorry about that. And then of course the government initiatives, which has been promoting a lot of translational research in today's time. Now we have heard this term quite often, translational research. And I'm not going to go into the definition of translational research, because I know that you are aware of it, but at least just to give an understanding of it. Many a times when we perform some kind of research, we tend to focus on what is the thing, what is the basic foundation that we want to look at, what is the basic biology or the physics or the chemistry behind what we are trying to understand. That is important, very important. But unless you have a question in your mind as to how this is going to help us in the bigger picture, in the sense that how it is going to translate and affect our lives, if you're not able to think about that question, the fundamental research that you are performing may not be as important or as reflective of the real world situation. So as I was introduced, the speaker did mention that I'm working in the field of regenerative medicine. Now if you look at the meaning of regenerative medicine, basically it has different pillars, which includes molecular biology. It includes a lot of use of biomaterials, and it also uses in the stem cells. So when you can cook up things together, then you get an understanding of regenerative medicine. And albeit when you start working with these, you definitely need to work on the fundamentals, the basics, which eventually can translate into the actual real world implications. So at this stage, I would not say that I have reached that stage where I can convert everything to the real world implications, but at least the starting point of the work that we have been doing in our lab at IIT Kharagpur has been the disease models that we want to focus upon. So before I go into my talk in actual, I'm sure many of you will be wondering about what has been my journey like. So was I, as I was introduced, that I have been, I'm a physician scientist. So I did my MBBS from Patna Medical College, but at that point of time, my primary focus was that I wanted to do clinics. I wanted to be a proper clinical practitioner. But during the course when I was studying, specifically in my second professional MBBS courses, pathology, there was a topic, and which is what I'm working on as well as one of the strong areas of my research, known as beta thalassemia. I'm not sure if you're aware of the disease, but it is a condition which involves congenital hemolytic anemia. And at that point of time, there was a situation where one of my family members got affected by that disease. And I was trying to understand the disease, and I found that there could be a way out for that disease. Of course, it's not something that is very easily or that can be done, but we may be able to think about an alternative way. And that was one of my drives which prompted me to look into research. So after finishing my MBBS, I went to UK for a short while, and I did some clinical attachments where I started understanding the basics of research, how it goes on in the Western world. Following which, I returned back to India, and I, in fact, started an MD pathology. But then again, when you go into a postgraduate program in medicine, it's most of the times you are doing the things which have already been established, the protocols that have been established in the labs. There is very little room for innovation. So I moved on to a program called Masters in Medical Science and Technology at IIT Kharagpur. And I got an opportunity, so it's a 3-year program. I got an opportunity to do an internship at the University of Toronto during my master's program. And as Professor Shah was mentioning about the importance of internships. So this particular internship that I did, of course, it was not an industry internship, but this is where I started my journey, per se, hands-on journey in research. So that's when I started understanding the intricacies of molecular biology and applications of molecular biology tools. As a clinician, I understood the diseases, the problems associated, but how you can actually start handling these diseases and look into the molecular world where nothing is visible, per se. So then I was becoming more and more interested in research, and so I did my PhD from Queensland University of Technology. And following that, when I came back to India, I did a brief stint with Philips as well. So that was my strong connect with the industry. But there again, I was not working in the field of regenerative medicine. Rather, I was working in the Philips healthcare and as a clinical specialist. So I was liaisoning between the industry and the academia. So there were a lot of clinical projects with ultrasonography where I used to be looking at the challenges associated, or if there are ways to actually find out other applications of the high-end ultrasonography equipments which Philips devices. So that was my contribution into development of new projects, or if there were some clinical projects which were ongoing, how I could contribute in terms of an alternative or a new form of application for that. But then my strong drive for regenerative medicine brought me back, and fortunately I got selected at IIT Kharagpur as a faculty member, and since then I have been at the School of Medical Science and Technology. Presently, actually, we have moved to a new building which has not been shown here. So my lab overall looks into a few different areas. As I mentioned, one of the areas is beta thalassemia. Basically, the idea is we focus from the pathophysiological manifestations, and why I say pathophysiological is because even in physiology, if you see, there are certain conditions which probably can help us get some cues which can be used in understanding the pathological mechanisms of diseases. So you start from the end point and then move towards the solutions. So I work on beta thalassemia, and as I said, regenerative medicine. So regeneration is the core area of my work. So even in beta thalassemia, which is a disease of hemoglobin, I'll talk about hemoglobin, what it is, and how we are trying to focus on regeneration in case of beta thalassemia. We are trying to actually reactivate a different form of hemoglobin where there is no problem, but nature, by its own reasons, has actually inhibited its manifestations in the postnatal life. So there is a theoretical possibility of improving that hemoglobin levels. And again, I have also been working on other diseases where there is inhibition of action. For example, osteoporosis. We are very well aware it's a condition where the bones become very brittle. So there is rarefaction of the bones. People become more prone to fractures. So if we can actually improvise these bones in some way, and if somebody is getting a fracture, we do put in implants. So if it is possible to improve and make the implants osseointegrated, or functional and structural integration with the bone in a much better way and a much faster way, can we do that? Then we are also focusing on other degenerative diseases like osteoarthritis, where biomaterials do play a very important role. I think you might be aware of different types of scaffolds which people use with drugs, without drugs, tissue engineering. 3D printing has come in a big way in today's world, where people are trying to use different ways to customize these implants. For example, people are trying to, so in case of osteoarthritis, if there is a specific deformity that you're trying to focus on, if it is possible to design that particular part of an individual and then replicate it using a tissue engineering approach and a 3D printing model and try to put it into the patient. And then, of course, wound healing is one of the core areas. And since I have been in clinical practice as well previously, so there is a strong drive as well to try and contribute somehow. So I have also contributed in case of point of care technologies as well. Now, this is something which I always want to focus, and I want to instill a thought in the students as to, and I think many of you might have seen this question. This is a 9-dot problem. So how do you connect it using just 4 lines? So most of the times we would try to do this and we will leave out the 2 dots. But the solution is usually this. And I don't know if there are any other solutions or not, but this is a solution which shows that many a times you might need to go out of the box thinking. So you have to come out of the box to think of a solution. So it's not always possible that wherever the problem lies, that is where the solution lies. That may not be the case. And why I am giving you this example is because I want to talk about beta thalassemia. I introduced you to an extent, but I want to now focus and show you what exactly we are working on. So some of you may be aware of what hemoglobin is. So very vaguely speaking, it's a pigment which is present on our red blood cells. So it is basically helping our exchange of oxygen and carbon dioxide in the body. So normal respiration happens because of the presence of hemoglobin. But what the problem lies is that it's a component which has heme, which is the iron component, and it has proteins surrounding it. So as you can see here, so there are what we see as, for example, many of you might have visited the pathology labs and got your hemoglobin tested. You get a specific value, and maybe based on the cutoff values, you are said that your hemoglobin levels are normal, or somebody is suffering from anemia, or maybe mild, moderate, or severe anemia. But there we are not looking into what is the type of anemia, or how can we correct it. So most common type is a nutritional deficiency anemia. So for example, people do not get enough of iron, or possibly folic acid, vitamin deficiency and all. So they can be easily rectified. So you just take supplements, or you take proper nutrition, and you will be okay. But at the same time, there are other types of anemia, and beta thalassemia is among one of them. Although it is classified as a rare disease in the world, but if you look at our country, it is kind of considered as the capital of thalassemia in the world. So around 3 to 5%, and specifically in the eastern part of the country, around 3 to 5% of us are having the gene mutations which can possibly cause thalassemia in offspring. So if 2 people who have these mutations have a baby, there is a high possibility that the baby is a thalassemia major. And it's a disease which is caused by a mutation in a particular chain, which is this, as I have shown the different types of hemoglobins. So this is the predominant form of hemoglobin. So more than 95% is the HbA form that is present. So among that, for example, you get a value of 15 gram per dl of hemoglobin. 95% is this. And there is an abnormality in this beta chain. So there is a mutation which causes the beta chain to not have that sufficient length. So it could be truncated, it could be completely absent, and this happens because of different types of mutations. So more than 200 different mutations have been identified so far. And of course, the first thought would be, how do we correct this mutation? So to correct this mutation, you would have to go back into the very basics and maybe try and correct it at the level of the in utero, within the fetus, in the early hematopoietic stem cells. because as you can understand, once the fetus starts growing, it's replicating in large numbers. So all the RBCs are having this type of hemoglobin. And for that matter, the number of RBCs, I think many of you will be aware, but for those who are not aware, we have around 5 to 6.5 million per cubic millimeter of blood. So that's huge numbers. You cannot go on correcting each and every RBC. Or for that matter, RBCs anyway do not have nucleus. You have to go to their precursors to correct it. So how can you do that? It's not theoretically possible, so to say. Of course, we are trying to develop mechanisms. For example, stem cell therapy you have heard about. So in stem cell therapy, what people try to do is that you ablate the original stem cells. So by inducing radiotherapy, you remove the defective stem cells, the hematopoietic stem cells, and you add in transplant normal stem cells from a donor. So that's one way out. But of course, you can imagine that it is fraught with its own challenges. Now I want to focus on the alternative form. So if you see, as I said, in adult life, more than 95% is this HbA form, which has this beta chain. When the baby is inside the uterus, this is not the predominant form. The predominant form is this one, HbF. So more than 95% is HbF. And it's a fully functional form of hemoglobin, which can carry appropriate amounts of oxygen and carbon dioxide. And we have absolutely normal, in most cases, we will have a normal chain of this gamma globin that you can see here. So the gene for this is perfectly normal, but because of a switchover mechanism that happens postnatally, just after birth, 4 to 6 weeks, there is a reason why this gets switched down and this HbA goes up. So instead of focusing on the abnormal form of hemoglobin, which is this beta globin chain, why can't we focus on the one which is normally existing, this delta chain? It's for some reason it is switched off. We say there's a switchover mechanism. So we are trying to focus on this alternative form and trying to understand why this switch off has happened and can we reverse this switch off. So this is where we are trying to work on, and as Professor Ravichandran was also discussing in today's morning about how these nucleotide analogs or how the nucleic acid related medications are being developed so far. So you can understand the implications of where we probably will be going in the future. So there is a class of small RNAs which are known as microRNAs, and they are indigenously present, inherently present in our body. So these small microRNAs have been found to be very important in gene regulation. Now what is gene regulation? So gene regulation is basically, you can imagine any function of the body, anything that we are doing or any protein that is expressed or any disease that is happening is because of a lot of genes working together, and some genes, proteins going up, some proteins going down. And all these proteins and genes, these omics that we talk about, they are under the control of some other kinds of molecules. So microRNAs being one of them. And yeah, I will skip this. So how do microRNAs work? Basically, if you see, if you have a word like complete and you add 2 letters IN in front of it, it does not become, it does not stop there, it becomes incomplete. So similarly, if a microRNA joins with its gene and there is a target specificity to it, then it decreases the gene expression. So it is not completely gone, but it decreases the gene expression. So it's a form of regulation. So you can either, it may completely reduce it as well, or it may partially reduce it. So accordingly, you can fine tune your mechanisms. So this is what we have been trying to work on. So I will not go into the details of these. As a part of our work, of course, a lot of in silico computer-based predictions have been going on. So we have developed different tools. For example, there is a mineralized tool which was developed by my student, and then we came up with a database of known validated targets of microRNAs. What in terms of experiments, what we have done so far is that what we have collaborations with NRS Medical College. So from there we have got a lot of samples from beta thalassemia patients' bloods, and from there we have collected the RNA, and we have compared it with normal people. So beta thalassemia people have been seen to have a relatively higher level of fetal hemoglobin, and normal people will have a normal level of hemoglobin. So we tried to compare their microRNA expressions, and we tried to understand which microRNAs are going up when the fetal hemoglobin levels are going up and which are going down. So based on that, we have come up with certain microRNAs which are upregulated and certain which are downregulated in patients having high fetal hemoglobin levels. And the present work that we are doing is related to that, where we are confirming the targets. So we do a lot of wet lab experiments in the lab. So we are trying to validate these targets. So microRNA, if you are introducing, so there are ways to introduce it. You can transfect the microRNA, a synthetic microRNA. You just put it into a cell, and then you see whether the level of the gene is going up or down. So we are developing those models as of now, and we are moving ahead. Again, I am skipping this, trying to keep it more general. And as I mentioned, the importance of basic research. So if you see this slide, this is again a different project that we are doing, again on beta thalassemia patients, where we are looking at some other genes. So there's a class of genes known as HLAs. HLAs, I don't know if you are aware, but these are like the molecules which are very important in case of infectious diseases. So what we try to see is whether there is a correlation between different specific HLA types. So if we have, let's say, 100 people in this room, we can categorize the people based on their HLA types as well. So HLAs, they can be a specific group having HLA A1, another group will be having A2, and so on and so forth. And so what we are trying to do right now is to categorize beta thalassemia patients as well, and trying to see if there is a correlation with these HLA patterns as well. And subsequent work is ongoing to look into whether there is an importance of this parasitic infections guiding the beta thalassemia patients. Yeah, so we have found some important results here, where we have seen when we categorize mild, moderate, and severe beta thalassemia patients, we do see some specific types of HLAs going up in some cases, and like we have been testing a few specific ones. So the other disease which we have worked on, and one of your faculty members here, Dr. Anisha Barik, was instrumental in this project. So we worked on osteoporosis. So osteoporosis is basically a disease where the total amount of bone osteoblasts and the osteocytes go down. It's a condition where the bones are very prone to fracture. And it's a condition which affects most of our old age people. More common in case of females, but it's not that males are not affected. So many of the male patients are also affected. So the chances of having a fracture in the old age because of deficiency of calcium, because of deficiency of multiple other factors, becomes very high, many folds high. And they do get fractures where they will have to be put in an implant. And as you would imagine, if you're placing an implant, that implant needs to integrate, so structurally and functionally join with the bone. So how do we achieve that? One way could be that we wait for an length of time. So we wait for months, days and months. In most cases, if you see there's a femoral fracture, so the fracture of the thigh bone, or if there is a fracture of any of the long bones, it takes a long time immobilization. So one way could be that if you can speeden up or hasten up this osseointegration property, so that the bone can bind to that implant much faster, superior osseointegration. Can that be achieved? People have tried in the past. Many different types of medications have been tried on the implants. Titanium implants are the most common implants that are used. So if you put those types of medications while placing the implants, then possibly the chances are that you might be able to enhance it. But of course, many of the drugs that have been used so far have had a lot of challenges. So we tried an alternative one, which is which is known as Tibolone. Now, this was a drug repurposing that we did. So this is a drug which is known as a selective estrogen receptor modulator. Previously, it was tried in clinical trials for in patients having postmenopausal symptoms. So as a part of the hormone replacement therapies. But during those trials, so they were being given oral therapies, and during those trials it was identified that some patients were developing cerebrovascular accidents, so strokes or maybe some cardiac manifestations were happening, and hence those clinical trials were discontinued over the time. And eventually the usage of this particular drug, which when it came, it was being considered as one of the best medications, but because of the trial efficacies showed these important side effects, so they discontinued its use. So we thought, why can't we try and repurpose it in the way that we do a localized release of Tibolone to improve osseointegration and osteogenic differentiation on titanium implant surfaces. So we did a lot of in vitro studies previously. So of course, we first did some in silico modeling studies where we tried to look into whether which type of genes should increase, which should go down, and based on those results we looked into how we can use this Tibolone on titanium implant surfaces. We want to coat the titanium implant surfaces, so we wanted to see whether they are actually helping in case of bone cell differentiation, so osteogenic differentiation happening or not. And in osteoporosis there is another type of cell which are known as the osteoclasts, which are very instrumental for osteoporosis. So the osteoclasts are basically cells which eat away the bone, so they become more active compared to normal cases, so they chew away the bone cells. So it has been identified that Tibolone can reduce the activity of osteoclasts, but it was not proven whether they can improve the activity of osteoblasts, the bone forming cells. So we looked into both, and we did find, of course, confirm that osteoclast activity was going down and osteoblastic activity was improving. So subsequently we did studies with coating, how we can coat the implant. So that was another challenge. So you just simply cannot put the Tibolone on the surface and expect it to be a sustained release, right? So you would have to have a material which can help it release over time in that localized vicinity of that fracture. So then it was tested with silk, and we did a lot of testing with this silk, and we identified the biocompatibility of the material that was developed. And eventually we studied in vitro whether osteogenic differentiation was happening. We did found good results over there. And yeah, this was a study which we did on an animal model. So where, of course, it took a while to optimize our studies, and Anisha very well remembers that. So initially, so this is where possibly our important role in terms of understanding anatomy came into play. We were lucky to have Dr. Rashmi Sinha, who is a gynecologist, who helped us in the initial phases trying to remove ovaries. So usually in these mice model, you need to remove the ovaries to induce osteoporosis. So that was the first learning, how do you develop these ovarectomy models. So and once we were able to establish that, then Anisha and other members of the lab, they were like replicating it very well, and then we had to place those implants. So that entire optimization process took a while, I think 6 to 8 months or so, and then we moved on. We did get really good results. So we did a lot of micro CT data, and we found that yes, our results were showing that it is helping in bone formation in a faster way on the titanium implant surfaces. Then, as I mentioned, we also have been working on some wound healing materials and osteoarthritis. So this is another work of one of my PhD students. So he's working on the silk and xanthan gum. So Xanthangum is a carbohydrate product which is identified in case of certain bacteria. So we are trying to see if we can actually join them together in some way. It could be a physical cross-linking, it could be a chemical cross-linking, and then help in translating it in the form of either a material that can help in osteoarthritis by improving chondrogenic differentiation, so cartilage formation can be enhanced, or another way, even by using them as wound dressing materials as a separate objective. So this is in the process of optimization. We have kind of narrowed down to certain things, and we are moving pretty well ahead with this. So yeah. And then, as I also mentioned about one of the clinical projects, which we did, this was in collaboration with our computer science department. Professor Joyanta Mukhopadhyay was very instrumental in it. So we developed a system, and now this is probably, like most of the things that I showed until now, they are mostly in that early developmental stages or early stages of the foundation being converted into trying to understand whether we can actually move to translation. But this is something which was more of a thing which integrated some of the existing tools and developing something which can help in developing a complete system. So here we were trying to develop a system for healthcare workers who are the first point of contact for our rural population. So I think many of you will be aware of the ASHA workers. So ASHA workers are the people who are the first point of contact for the rural women and children, and even for everyone else as well. But for pregnant mothers, most of the times it is like, if you are looking into the interiors of our villages, it is not possible for them to reach out to the doctors or even to hospitals at the very first go. So ASHA workers are the first point of contact, and most of the times the ASHA workers are like high school educated. So although they are willing to help, although they have been trained, but they usually are not able to identify the red flags where they need to send the patient immediately to the doctor or immediately to the nearest hospital. So what we tried to develop was a system which would look into certain simple things, for example, the hemoglobin levels or the blood pressure level. Now blood pressure, I am sure you have seen these automated blood pressure monitoring systems. You just have to tie the cuff and it gives up the values. But again, the implications of those values are not always very easy to understand. And specifically in case of pregnant women, it is not always very easy to understand depending on other conditions which the person will be having. So hemoglobin levels, blood pressure values, AccuCheck, blood pressures, blood sugar monitoring, that is also important, or looking into if there is protein which is present in urine. In normal conditions, you will not get any protein in urine, but there is a condition known as eclampsia. So the seizures which might happen, which might actually affect pregnant women, and the baby can be dead, or even the pregnant women may have a lot of complications associated. So if somebody is prone to developing that, so how do we recognize that? So we integrated some of the existing tools, and in addition we also developed an indigenously developed in our lab system which can detect the hemoglobin levels by just a single drop of blood, and that same system can also be used for the process of looking into whether there is protein and how much protein is present. If there is protein, how much is the amount of protein which may be present in the urine. So this was we have already filed the patent for this, and very recently we also got a paper accepted for this. So this is this is one of the earlier prototypes that I'm showing, but this is the kind of the black box that you can see here, where you just put one drop of blood and it gives you a value. And this entire system will come up with a decision support algorithm. So it comes up with the values, all the values together, and this has been developed in conjunction with clinicians who have been working in the field. So they have assigned different scores to different levels of each of the conditions. For example, 180 by 100 blood pressure is a very high level of blood pressure. that along with very high levels of blood sugar, that would definitely mean a red alert. Or for that matter, somebody having a relatively less amount of blood pressure, but maybe a high sugar level may not be that dangerous. So it probably will come up the yellow range. So there probably you can wait for another visit. So it gives a system with a score which the ASHA worker can easily understand based on the clinician's methodology, and according to that the triaging can be easily done. So which patient has to immediately visit the doctor, which patient has to immediately like you need to go immediately after a few days, or which patient can stay at home and maybe look at it at a later date. So this is also another work, but of course this is not my forte. My forte was to look into the clinical understanding of this. The entire image processing system and all were developed from the computer science department. Sorry. Yeah, so I mean, of course, none of this work that I showed today would have been possible without the students and my staff members in the lab that I have. And these are some of the pictures that we have from our lab. I wanted to have some more pictures. Unfortunately, we couldn't get it. But yes, thanks to my students, my first two PhD students, Shankho and Anisha, and the students who are still there, Prasanna, Motiur, Urchita, Shantani, Mandrita, Lopa, Shatrupa, Deblina, then the staffs, Adari and Shuman and Shukhen. Yeah, and of course, none of this work would have been possible without the funding bodies, right? So thanks to each and every funding agency. So as you can see, most of the funding has come from government funding agencies. There are now, as I mentioned, there are some funding bodies which are like working together, where an academic partner and an industry partner needs to go hand in hand. So the work which I showed you regarding that device, the Korok, was actually started with it started like a joint collaboration with a startup company. Unfortunately, the company had to bootstrap and then they had to close down, and eventually we took it forward. So they had their own limitations for the reasons which are not very clear, but they had to close down. But yes, we were able to drive the project, and luckily we have come up with a good solution as of now, which with the patent pending right now. So yeah, and I think in my introduction you also mentioned about INEAS. So the last slide would be to talk about the Indian National Young Academy of Sciences, for which I am a National Core Committee member. So I think just like in corporate, we have this corporate social responsibility, which is like a part where a part of the profits need to go to the society. Similarly, as scientists, we also have certain responsibilities to the society, where we not only are focusing our work to either going to the industry or for our own satisfaction, rather it should be something that can be translated to the society. So now DST and other ministries have also come up with this term called SSR, Scientific Social Responsibility. So INEAS is one of the academies. It's a young academy which is doing venerable work in case of connecting science with society. So I'm lucky to be a part of that organization, and these are just some of the glimpses of the kind of science outreach activities that we do. So recently, even as one of the projects, sorry, not a project, as one of the events which we did through INEAS was known as this, one was for rural science teachers. So it was called Are You Set Up? So we did a training for school teachers. So Professor Shuman Chakraborty was one of the speakers. I don't know if you are aware. He got the Infosys Prize this year. So Professor Shuman Chakraborty does magnificent work in the field of point of care technologies. And the way you can communicate science to the society is not by using scientific jargon. So many a times we tend to be bogged down by the terminology that we use. Of course, today I was using because I was in front of an expert audience as well. So you all students of science. So I know that when I speak certain terminologies, you would be understanding. But if you are trying to connect with the society, it's not that everybody will be understanding these terminologies. So you need to be going down to the level where people can understand what you're talking about. And why this is important? The government, the industry, everybody is funding our work. So there is a responsibility from our part as well as to make people understand what we are doing, why is this important, and how this is going to impact lives. So when I was doing my PhD at Queensland University of Technology, we used to have a conference, in-house conference, and there one of the statements we had to mention was real world implications. So what is the real world implication of the work that you are doing? So as students, whenever you are involved in certain kinds of projects, it is my earnest request is to first ask this question to yourself, that what is the implication of this activity that you are doing? How is this going to impact in terms of the contribution to the society at large? It may not be that you always go for translational research or that you are coming up with a device, but at least even if you're going into the fundamentals, how is this going to impact the actual thing? Even today morning's talk, Professor Ravichandran mentioned about the importance of fundamental research. So do focus on fundamentals, but with a thought in mind as to how do you translate it to the actual real world. And with this, I would like to end my talk for today, and I'll be happy to take up questions. Thank you.
So students,
So again the same thing. I think because this one is working, you need to turn it on.
hello. Okay, so students, if you have any question, you can ask.
Yes, please. So I think there are some lower animals possibly, but I may be mistaken. I have to check it. But I think there is some animal which, but yeah, maybe I'm mistaken. Possibly, I'm not sure. So yeah, zoology is not my focus. So maybe I'm mistaken, but if I think there is some animal existing. Yeah, possible. I think what you're saying, probably camel. I don't know. Yeah, so it's possible. But the nucleus is just removed before the mature RBC is produced. So even in the reticulocyte state, you still have some amount of nucleus which is still present, even in humans. So, and the entire differentiation is when the final RBC is produced. So that means that the entire thing is being guided by the nuclear machinery for sure. So although what you are seeing as the end product is the one which does not have a nucleus, yet the end product is because of the nuclear machinery. So we cannot negate its implications, and we should not negate the things that we have been working on are based on the nucleated cells that are present. Yeah.
Okay, so any other question from the audience? Okay, so thank you, sir, for such an enlightening session. Now I would request Professor Dr. Bishwajit Das to kindly come up on the stage and present the token of appreciation to our speaker. Thank you, sir. Thank you so much.
Thank you very much.
शुभम कुरुत्वम कल्याणम माराग्यम धनसंपदा शत्रुवृद्धि विनाशाय संध्यादीपम नमस्तुते. The Ardent lamp enlightens us. It reduces the negativity, the darkness. like this. Thank you, sir. Thank you so much for being present here. You enlightened us through your expertise and knowledge. I hope in future from Adamas University we can collaborate with us in any kind of projects regarding this regenerative medicines re-engineering process. Thank you so much. Cheers. Thank you. Thank you, sir.
So this one, pen drive. Just end করবে. End. So we have come to the end of the first day seminar session. You will, so hope all of you will join soon again tomorrow sharp at 10 a.m. at AU1 auditorium. I repeat, for the next session of seminar, please join tomorrow at AU1 auditorium sharp at 10 a.m. Thank you all.